Okay, everybody, welcome to Chapter 7, dealing with the nervous system. This is going to be one of the more complicated chapters in the book because the nervous system has a large role in maintaining homeostasis in the body, and it's somewhat more complicated than some of the other organ systems that we're going to talk about this term. So, what is the job of the nervous system? And one of its major jobs is to respond to the environment, and this falls under the role of sensory input. Essentially, it gathers information in order to monitor changes that occur outside and inside the body. These changes are what we call stimuli. Integration is important in order to process and interpret the sensory input and decide if action needs to be taken in order to ensure further fitness and survival. And motor output is a response to these integrated stimuli, and the response activates muscles or glands. And so if we look here at a diagram showing you the way that information flows into and out of the nervous system, you can see that the sensory input comes from special structures that are embedded in the body's tissues that are called sensory receptors. And some of these receptors are very specialized. For instance, the receptors that are in the back of the eye, shown here, are capable of responding to light. They can interpret color, and they can interpret the intensity of light, and they can transform that information into an electrical signal that'll eventually be sent through sensory nerves back to the brain, where the brain will take this information along with other information that's coming in to the brain at the same time and decide on a course of action. That course of action will then be sent from the brain to the brainstem and then down to the spinal cord and out through the peripheral nerves to the effector, which will be perhaps a group of muscles that will contract in order to perform some kind of action, or it could be any of a number of other integrated responses. The important thing to understand about these sensory inputs and these motor outputs is that they're electrical in nature. The brain can't directly understand stimuli such as light, pressure, sound, heat, cold, and so on in a direct sense. It has to be turned into an electrical pattern that can be sent back to the brain, and what it'll do is stimulate specific regions of the brain that can then put a category on those different types of information, and then using that categorized information form a plan of action. The plan of action is a series of electrical signals that get sent from the brain to the brainstem to the spinal cord and out through the peripheral nerves, and those will hit effector organs. They could be muscles, they could be glands, they could be other tissues, and some sort of action will be carried out. Okay, so we go from physical stimulus to electrical information. In the brain, the electrical information gets integrated. We form a plan in the form of electrical patterns that get sent from the brain to the brainstem to the spinal cord out to the peripheral nerves. This electrical information reaches the effectors, and that electrical information causes the effectors to do something. It might cause a muscle to contract, it might cause a gland to secrete, it might cause any of a number of other physical responses. So when we talk about the central nervous system, we're referring to a couple of very important organs, and those include the brain and the spinal cord. Their function is to integrate sensory stimuli and then send commands out the central nervous system to the peripheral nerves to the effector organs. This is what we mean when we say it's capable of issuing outgoing instructions. Again, these instructions are electrical in nature. The peripheral nervous system is everything outside the brain, the brainstem, and the spinal cord. It consists of the nerves that extend from the brain and the spinal cord to their target organs. Spinal nerves carry impulses to and from the spinal cord, while cranial nerves carry impulses to and from the brain. And their function is to serve as a communication line among the sensory organs, the brain, the spinal cord, and the glands and the muscles. So this is the way the electrical information gets out of the brain and the spinal cord and travels to the effectors, and it's also the way that the information comes into the central nervous system and gets eventually sent back to the brainstem and the brain, where it's integrated, it's categorized, and then we decide what it is we want to do about the sensory input. So there's two divisions of the peripheral nervous system that we need to be aware of. The sensory, or the afferent division, consists of nerve fibers that carry information to the central nervous system, while the efferent division are nerve fibers that carry impulses away from the central nervous system. So two different directions here. And this chart kind of shows you how the central and the peripheral nervous system operate. So we start with the central nervous system. We could start, say, with the central nervous system receiving information from the sense organs. So we would start with some sort of specialized receptor, let's say the retina in the back of the eye, which responds to light and color, changing that information into electrical information. That electrical information travels along the afferent division through the peripheral nerves and eventually makes it to the spinal nerves, which enter the spinal cord and then carry that up to the brainstem and the brain. And what happens next is we decide what we want to do about that electrical information. We interpret that electrical information as light and color because it stimulates the appropriate part of the brain that is able to deal with visual information. And then we form a plan of action. The plan of action is generated in the brain, is sent down to the brainstem and to the spinal cord, and then it exits the central nervous system through the peripheral nerves, which then carry that information via the efferent division to any of a number of different locations. If it carries it to muscle, okay, this is what we would call the somatic motor system, and this is the type of motor information that we're in control of. Our conscious thought directs these actions. Or the motor division can also carry information that comes not so much from the brain, but more from the brainstem, that control what we call our autonomic functions. These are body functions that keep us alive that we don't have to dedicate conscious thought to controlling. And this includes the innervation of cardiac and smooth muscle and the innervation of glands in order to cause them, say, to secrete at appropriate times. There's two divisions in the autonomic nervous system. The parasympathetic division, which has the nickname the rest and digest division, and the sympathetic division, which is called the fight or flight division. And they describe the unconscious physical responses that you would have faced with a certain set of circumstances. For instance, the sympathetic nervous system would be activated if we were to find ourselves in a situation that we perceived as dangerous. Okay, and what might happen as a result of sympathetic signals being sent to muscles and glands and tissues throughout the body is that our heart rate would increase, our respiratory rate would go up, our pupils would dilate, and our liver would begin to generate glucose from glycogen in order to provide fuel in the blood in order for us to take action against our perceived danger. And we'd also have increased blood pressure. The body would attempt to try and retain more of its water. As a result, urine volume would go down, blood volume would go up, and blood pressure would go up. And again, that's an effort to try and get as much blood to the skeletal muscle as possible so that we can respond to this perceived dangerous situation. And then what would happen is we would take action to extract ourselves from danger. The rest and digest division is responsible for um, the body's state when it's essentially processing nutrients and replenishing fuel supplies. And so typical parasympathetic responses include increased GI motility, pupillary constriction, blood pressure drops, heart rate and respiratory rate drop. All these are the kinds of responses we would expect to see, say, after finishing a large meal and relaxing on the couch. Okay, hence the nickname the rest and digest division. Now we have functional classifications for the peripheral nervous system. The motor or efferent division has two subdivisions. The somatic nervous system controls our voluntary activity, so most of its targets are skeletal muscle. And the autonomic nervous system is our involuntary response to stimulus, and it automatically controls smooth and cardiac muscle and glandular tissue throughout the body. And as we stated earlier, it's further divided into the sympathetic and parasympathetic nervous system. Nervous tissue, if we take a look at it, is made up of two classes of cells. The neurons, which send and receive the electrical information and process it, and the glial cells, which you could think of as the personal assistants to the nervous system. And they take care of all kinds of very important functions that the neuron really doesn't have time to deal with, because electrical information needs to be processed so rapidly that the nerve cell really doesn't have time to do things such as gather nutrients, protect itself against damage, remove waste products, and insulate and protect the neurons. The glial cells do all of these different functions, and as a result, the nerve cell is free to deal with the onslaught of electrical information that run through it thousands of times a second, while these other cells, these glial cells, take care of these other housekeeping functions. So the first glial cell we want to talk about is the astrocyte. The reason astrocytes are called astrocytes is because they are star-shaped. They brace the neurons and they form a barrier between the capillaries and the neurons, and they control the chemical environment of the brain. And essentially what they do is they extract nutrients from the circulatory system and provide those nutrients to the nerve cells, and they also help remove waste products. So you could think of them as the personal shoppers and garbage disposal of the nervous system. One very important thing to realize about nerve tissue is because of these astrocytes, the formed elements of blood never come into direct contact with nervous tissue unless there's been some sort of trauma, some kind of damage due to accident. As a result, nerve tissue has what we call blood-brain barrier, where we don't have to worry about the phenomenon of immune rejection in the event we were to transplant nerve tissue from one individual to another, because of this barrier that's set up by these glial cells. Okay, there's other glial cells that we'll talk about that further isolate the tissue of the peripheral and the central nervous system from the formed elements of blood as well. But here you can see how these star-shaped cells are capable of extracting nutrients from the blood and providing them to the nerve tissue. Now, microglia you could think of as the emergency med techs of the nervous system, and what they do is they dispose of debris and they clean up damage. They have a spider-like appearance, and they're seen in areas of the nervous system that have suffered trauma. So again, the nerve cell doesn't have to worry about defending itself against an onslaught of, say, a pathogen or a disease, because these microglia are always nearby, ready to take care of those problems. Now, ependymal cells are special. Ependymal cells line cavities of the brain and the spinal cord, and the cilia assist with the circulation of a very special kind of fluid called cerebrospinal fluid, which bathes the tissue of the central nervous system and serves as a homeostatic environment for the nerve cells to carry out their activity. The temperature and the pH and the ion concentration are carefully controlled, this fluid is essentially a filtrate of blood that helps maintain homeostatic conditions in and around the neurons. And so this is constantly being filtered from the blood. It's bathing the brain and the spinal cord, and it eventually rejoins the circulatory system in very special structures located just superficial to the brain, called arachnoid granulations. And there the cerebrospinal fluid will diffuse back into the circulatory system and rejoin the general circulation. These ependymal cells also establish further something called the blood-brain barrier. And again, what this speaks to is the fact that the formed elements of blood never come into direct contact with nervous tissue, in part because of the barrier set up by these glial cells. Now, oligodendrocytes are found in the central nervous system, and their job is to insulate the nerve fibers so that electrical signals get from their point of origin to their intended target. They wrap around the nerve fibers in the central nervous system, and they produce a very special lipid-filled substance that's called myelin, which acts kind of like the rubber insulation around a wire, and it keeps the electrical signal from leaking out and not getting to its intended target. So this protects and it insulates the electrical signal that runs through the neurons. These long strands here that extend out from the nerve cells are called axons, and they carry the electrical signal away from the nerve cell body and to either the next neuron in the chain or to a target tissue of some kind. Here we can see their counterpart in the peripheral nervous system, and these are called satellite cells, and what they do is they protect neuron cell bodies, okay, by wrapping around them and insulating them from damage. And then the Schwann cells, which you can see wrapped around the processes that extend out from the nerve cell body, and they do the same thing that the oligodendrocytes do in the central nervous system, which is insulate the electrical signal and make certain that it finds its target. So you can see here each one of these little blue spaces represents a single Schwann cell, and you've got a series of them wrapped around the axon of this particular neuron. And what this does is it not only insulates the nerve signal so that it reaches its intended target, but as we're going to find out later on in the chapter, these little gaps between the Schwann cells serve as trip points for electrical signals to move very rapidly from one end of the axon to the other until they get all the way to the end of the axon, where they'll trigger the release of a chemical called a neurotransmitter, which will diffuse across a tiny gap between one nerve cell and its target, whether that be another neuron or some other type of tissue. The neurotransmitter will then bind to receptors on the target cell membrane, and that'll produce a voltage change in that membrane that will lead to some sort of electrical activity. Okay, in the case of muscle, it might lead to muscle contraction. In the case of a nerve cell, it could lead to that nerve cell firing, just as a couple of examples. Neurons are nerve cells, and they're specialized to transmit electrical information. The major regions of the neuron include the cell body, which contains the nucleus and the metabolic center of the cell, and the processes that radiate out from the cell body, which are called dendrites, which are short processes that radiate directly off of a part of the cell body that's called the soma, and they are basically receivers of electrical information, and then a very long process that's called the axon, which is the sender of the electrical information. It conveys the electrical information away from the cell body and ultimately to the next cell in the nerve pathway. The cell body contains unusual structures called Nissl bodies, which are a specialized rough endoplasmic reticulum that contains a dark pigment that's the result of metabolic breakdown products that have to be stored inside the nerve cell because they don't have any other avenue to be removed from the cytoplasm. Neurofibrils are a cytoskeletal element that are inside the nerve cell. They maintain the cell shape. And the soma of the cell contains a nucleus with a very large nucleolus, because there's a lot of protein synthesis that's going on inside neurons, as well as a lot of neurotransmitter synthesis, and so we have to have a lot of protein building machinery. And remember that the nucleolus is the site of the synthesis of ribosomal RNA, which is one of the structural components of the ribosome. Remember that the ribosomes are the protein builders in the cell. There are processes that are outside the cell body. The dendrites conduct impulses towards the cell body. A neuron may have hundreds of dendrites, and you could think of them like antennae, and what they do is they receive electrical information from other cells, maybe other neurons, maybe other types of tissue entirely. The axons conduct the impulse away from the cell body. Neurons have only one axon that comes from the cell body at the axon hillock, and here you can see a diagram of a typical motor neuron. This is the soma, these are the Nissl bodies, and this is the nucleus with the prominent nucleolus, and these little projections here, these are the dendrites. These receive the electrical information. The electrical information then sort of coalesces on the surface of the soma, and then eventually what will happen is that all that electrical information will reach a very critical junction in the neuron called the axon hillock, which is the gateway to the axon. And when that happens, if we reach a certain critical voltage as a result of all these electrical signals that are coming in from all directions and hitting the nerve cell, what will happen is that the axon hillock will start a series of events that will result in an electrical signal called an action potential being sent away from the soma and all the way down to the end of the axon, where it'll hit eventually these little sort of bulb-like processes on the end of the axon, which are called the synaptic boutons or the axon terminals. When that happens, calcium gates will open up, and the calcium will move into the axon terminal, and that will cause little membrane-filled vesicles, basically membrane wrapped around neurotransmitter, to fuse with the ends of these synaptic terminals and release the neurotransmitter into the gap. And what will happen next is the neurotransmitter will flow across this tiny gap and bind to the membrane of the target, and that will produce some sort of electrical response in the target. The target may be another neuron, the target may be a gland, it may be a muscle. It all depends, okay, where we are in the nerve pathway. You can see here in this micrograph a look at a neuron cell body and the dendrites and what looks like the axon coming off the end right there. Okay, looking at the neuron, we notice that there are several processes that radiate out from the soma, which is the neuron cell body. The longest is the axon, which ends in an axon terminal that contains vesicles that enclose neurotransmitters, which are chemicals that carry electrical signals across a physical gap between a nerve cell and its target, which could be another nerve cell, it could be a muscle cell, it could be a glandular structure, a tissue of some kind. The targets can vary. The axon terminals are separated from the next neuron by a gap, which is called the synaptic cleft, a physical space between two neurons or between a neuron and its target. The synapse is the junction between these two nerves, and what happens at this junction is that when a nerve signal reaches the end of a nerve cell, it will trigger an influx of calcium into the axon terminal. As the calcium moves in to the end of the axon, it causes these vesicles that contain the neurotransmitters to fuse with the axon terminal and release the neurotransmitter into the gap. And then they'll flow across a short physical space and bind to the postsynaptic membrane, and that will produce a voltage change in the postsynaptic membrane, which will cause the target to respond. Now, if it's another neuron, this voltage change may cause the neuron to tend more towards firing, or it may cause it to tend to be less likely to fire. It all depends on the nature of the neurotransmitter that's released. In the neuromuscular junction, the gap between the muscle cell and the nerve cell, the motor neuron, the neurotransmitter is acetylcholine, and that is always an excitatory neurotransmitter. It always causes the muscle cell to fire, and that results in a muscle contraction. Now, the myelin sheath is an insulating structure that keeps the electrical signal from not reaching its intended target. It's a whitish fatty material that covers the axon. Schwann cells make the myelin sheaths in sort of a jelly roll sort of fashion. They wrap around the axons at predetermined points in the peripheral nervous system. And what they form in between where the Schwann cells are located are what we call nodes of Rabnier, gaps in the myelin sheath that run along the axon. And they represent areas where the electrical signal can hop from the gap between two Schwann cells to the gap between the next two Schwann cells as it goes very rapidly down the axon, away from the soma and towards the axon terminal. And this is a type of nerve conduction that we call saltatory conduction. Saltatory conduction means to leap or to hop from one location to the next. Now, oligodendrocytes perform this function in the central nervous system. They make the myelin sheaths that surround the axons of the central nervous system, and they're associated with white matter in the central nervous system. In the spinal cord, the white matter tends to be surrounding the periphery of the spinal cord with the gray matter inside. And in the brain, the white matter tends to be deep inside the brain with a surface coating of gray matter. Now you might say, okay, well, you've told us what the white matter is. What is the gray matter? The gray matter are collections of unmyelinated fibers and cell bodies. And so when you see white matter and gray matter in nervous tissue, you should immediately think of two different types of processes that happen to electrical information. In white matter, what's happening is that the signal is being sent from one place to another, whereas in gray matter, what's happening is that the signal is being processed and modified. So when you see gray matter, think cell bodies. When you see white matter, think myelinated axons, and that will tell you the difference between nerve cell signal transmission and nerve cell signal processing. Most neuron cell bodies are found in the central nervous system. The gray matter are, again, the collections of the unmyelinated fibers and cell bodies. And then sometimes we'll refer to a location in the brain or the spinal cord or the brainstem as a nucleus. And this is really the third use of the word nucleus in a course like this. And here it means clusters of tissue that perform a particular function. The first use of the word nucleus was as the nucleus of an atom, where the protons and neutrons are contained in matter. The second term where nucleus was used was in the nucleus of a cell, where the genetic material, DNA, is contained. And this is the third use of the word nucleus, and it refers to a location of tissue that's dedicated to a particular task. Ganglia are collections of cell bodies outside the central nervous system, and tracts are bundles of nerve fibers within the central nervous system, and they tend to be white matter. Nerves are bundles of nerve fibers in the peripheral nervous system, and white matter are collections of myelinated fibers. Now, when we think of tracts and nerves, we have to consider that when you look at a particular tract or you look at a particular nerve, depending on where it's located, the information can either be afferent, meaning that it's coming into the central nervous system from the outside, or it can be efferent, meaning it's leaving the central nervous system and heading to a target, or sometimes a single nerve or a single tract can carry information going in two directions, and that's what we call mixed. We can have mixed nerves. Sometimes we can have mixed tracts, okay, that will send information in different directions. And the reason that this is possible is because of the insulation that's provided by the myelin and also by the connective tissue that isolates nerve cells from each other in structures like the brain and the spinal cord and in the peripheral nerves. Okay, afferent neurons carry impulses from the sensory receptors into the central nervous system. You've got cutaneous sense organs that are located in the skin, and they tell you things about your outside environment, such as temperature. They provide a pain signal. They allow you to respond to things like pressure and light touch, irritation, such as inflammation, itching. Basically, your general senses come from these organs. Proprioreceptors detect stretch or tension, and we find a lot of these located in muscles and in joints. And this is your ability to perceive your body's location in physical space, and that information is sent up to the brain so that we can deal with it and adjust to our changing situation, right? As we're moving, for instance, or as say we're falling or walking or whatever, that information gets sent up to the brain, and the brain is able to integrate it, to categorize it, and then the motor portion of the brain is able to respond appropriately to that information and send the signal in terms of what to do in the opposite direction, going from the brain to the brainstem to the spinal cord, and then out to the peripheral nerves to the muscles, and then we adjust what it is we're doing to suit our needs at the time. Motor neurons carry impulses from the central nervous system to the viscera, the muscles, or the glands, and so that is outgoing information. That information actually starts in the brain, in an area called the frontal lobe, where we form plans of action. We then send those plans of action, those electrical signals, to an area of the brain called the precentral gyrus, which is a ridge of tissue that runs across the surface of the brain like a hair barrette does, if you've ever seen women or young girls wear those to keep their hair back. It's a ridge of tissue that sits just in front of a groove in the brain called the central sulcus. And what happens in this ridge of tissue called the precentral gyrus is that the signals to carry out the motor activity are sent from there to the brainstem and to the spinal cord, and from there to the peripheral nerves, and from there to the muscles, and that's how we're able to carry out a plan of action. Interneurons are found in neural pathways in the central nervous system, and they connect sensory and motor neurons to each other. And so you can see here, this is a cross-section of the spinal cord. This is the back of the spinal cord. This is the front of the spinal cord. This is white matter, so this is myelinated axons out here. And this is gray matter. This is primarily neuron cell bodies and unmyelinated cell fibers. This is the central canal. This is an opening through which cerebrospinal fluid flows through the center of the spinal cord, which is about the width of your little finger. And you've got a posterior groove in here, this little indentation, which is called the this is a sulcus, okay, a deep groove in the back of the spinal cord. This is the dorsal groove, or the dorsal sulcus, and this is the ventral groove, or ventral sulcus. And you can see that the groove is much deeper in the back than in the front. In addition, if you look at the back of the spinal cord and you look at the spinal nerves that come in to the back of the spinal cord, you see a cluster of nerve cell bodies called the dorsal root ganglion, which is always associated with the back of the spinal cord. So this is one of the ways that you can tell back from front, is to look for the dorsal root ganglion when you look at a figure like this. So this is the back, this is the front. What happens at the level of the spinal cord is that sensory information comes in through afferent neurons, through these spinal nerves, through the dorsal root ganglion, and is transmitted through the dorsal root, this is the dorsal root, into now what we call the dorsal horn of the gray matter of the spinal cord. That information will then be sent up to the brainstem and to the brain, where ultimately a plan of action will result, and then information coming in the other direction from the brain to the brainstem to the spinal cord will eventually reach the appropriate level of the spinal cord for the information to exit, and it will come out, the motor information, through what we call the ventral root. This is the ventral root. These are motor neurons, and what they do is they carry outgoing information all the way to the target. Okay, now the target could be a gland, it could be a muscle, it could be other types of tissue. But just remember, information comes in the back, the afferent neurons, and motor information comes out the front through the efferent neurons or motor neurons here. And then there's integration of the information in the gray matter of the spinal cord at the level of what we call interneurons, which allow communication between nerve cells of what we call ascending and descending tracts. Ascending tracts head up the spinal cord towards the brainstem and the brain and terminate somewhere either in the brainstem or the brain, and descending tracts go in the opposite direction from the brain to the brainstem and then down to whatever level of the spinal cord they're fit to exit. Okay, so these interneurons are a way of communicating between neurons. In some of the very simplest neural pathways called reflexes, what happens is that sensory information goes directly through an afferent neuron and hits an interneuron, which synapses directly onto a motor neuron, and that results in a stereotypical behavior, such as if you put your hand on a hot stove, right, and the pain signal comes in through the afferent division and immediately synapses on an interneuron, which hits a motor neuron, which goes out the efferent division, and you yank your hand straight up from the hot stove before your brain really gets the message that you did something stupid. It gets the information, but it gets the information kind of after the fact. And when we talk a little bit more about reflexes, you can appreciate how the incoming and outgoing information are coordinated at the level of the spinal cord, the brainstem, and the brain in order to carry these activities out and keep you alive, essentially. Everything outside the brain and brainstem and spinal cord is what we call the peripheral nervous system. Okay, so this would all be peripheral nervous system tissue, and the brain, the brainstem, and the spinal cord are all central nervous system tissue. So this would all be central nervous system tissue. Again, the gray matter containing the cell bodies, the white matter containing the myelinated axons, which represent either the ascending tracts heading up towards the brain, or the descending tracts heading down from the brain to the spinal cord and then out through the peripheral nervous system to the target. Okay. You can see here some examples of different types of nerve endings and what they detect. Free nerve endings in the dermis detect pain. You can see them here. They essentially have no modifications to their ends. And what happens when a painful stimulus is encountered, such as a puncture or a laceration or an abrasion, is that they fire, and then they send pain information up ascending pathways from the spinal cord to the brain, and we respond appropriately. Meissner's corpuscles are touch receptors. You can see that we've got these layers of connective tissue surrounding the free nerve ending, and this modifies this type of nerve ending to respond to light touch through changes in its shape. These are what are called mechanoreceptors. As they change their shape, they produce an electrical signal. That electrical signal gets sent to the nerve, from there to the spinal cord, and from there to the brainstem, and from there to the brain. And we perceive light touch. Lamellar corpuscles are modified for a response to deep pressure. They're a little bit deeper in the skin, and they have a slightly different modification of their nerve endings. They only deform in response to very, very deep pressure, and they send that information back up to the brain, and we respond appropriately. The Golgi tendon organs are isolated inside tendons and inside muscle fibers, and what they do is they provide our body with its sense of physical position in space. They're sort of a fail-safe along with the muscle spindle over here, so that muscles don't contract in an unexpected way and produce damage, such as pulling bone straight off of its attachment to the tendon. That's called an avulsion, or causing the muscle itself to rupture. What these tendon organs and these muscle spindles do is they inform the brain of your motor activity as it's happening, so that we can make proper physical adjustments in order for our muscles to anticipate what's coming next. And we'll get into exactly how these tendon organs and muscle spindles work a little bit later on. But what you see here is that in each case you have modified a nerve ending so that a physical stimulus gets turned into an electrical stimulus, and it's the electrical stimulus that allows the brain to perceive what's going on in the outside world. The brain has no ability to directly respond to physical stimuli. It's got no receptors in it. It only responds to electrical information, and this is how we change physical information from electrical information and get it up to the brain so that it can form a plan of action. Now, neurons come in many shapes and sizes. The first neuron we want to look at is the multipolar neuron. It's called multipolar because it has many extensions radiating out from the soma. This is the soma, or cell body, and you can see here that we've got dendrites, which are like the cell's antenna, that are responsive to electrical signals that are produced as a result of binding to neurotransmitters and generating voltage changes that may cause the nerve cell to generate an action potential. And then you've got the axon, which is this long projection that heads away from the cell body and carries the electrical signal from what we call the axon hillock, this little area right here, all the way to the end of the axon, where it ends at the axon terminal and results in the release of neurotransmitter, which will carry the electrical signal to the next cell in the nerve pathway, whether that be another neuron or whether that be some other type of target tissue. This is one of the most common types of neurons you'll find in the body. Motor neurons look a lot like this. They have these long myelinated axons, and they're some of the fastest neurons in the body. They can carry electrical signals at speeds of close to 200 miles an hour from one region of the body to the next. And it's this saltatory conduction that allows that to happen. Something to keep in mind is that electrical signals move very rapidly through nerve cells. The slowest part of any nerve pathway is the part where the neurotransmitter is secreted from the axon terminal and floats across the tiny gap to bind to the postsynaptic membrane and generate an electrical response in the target. So the more synapses you have in a nerve pathway, the slower it's going to be. The fewer synapses you have, the more rapid it's going to be. Bipolar neurons have one axon and one dendrite. You can see the dendrite here, the cell body or soma, and the axon. And they're located in special sense organs like the nose and the eye. They tend to be relatively rare in adults. And again, the nerve signal goes from dendrite to cell body to axon. So if we were To draw that, the nerve signal would go this way, okay, and eventually would get to the end of the cell, and at this point calcium would rush into the axon terminal and neurotransmitter would be secreted into the synapse. Little dots here representing neurotransmitter, not the greatest artist, and then they would bind to the membrane on the target cell and produce an electrical change that may result in that particular target cell doing something. Unipolar neurons have a short single process that leaves the cell body, which can essentially become an axon that looks essentially fused to the cell body through a single short process. So you can see dendrites up here receiving electrical signals and then transmitting them very rapidly through this long myelinated process, essentially almost bypassing the cell body and heading straight down to the end of the axon terminal, where again it'll result in the release of neurotransmitter. So again, if we were to draw this, the direction of the signal would be this way, okay, coming in here and heading out there. We find these sensory neurons in peripheral nervous system ganglia. Some of the functional properties of neurons include irritability, which is the ability to respond to a physical or an electrical stimulus, and conductivity, which is the ability to transmit an electrical impulse. And so remember that we talked in the first week of class about the fact that the reason that cells have these special properties is because they have a particular job to do. And in the case of the nerve cell, it has been modified to send and receive electrical information. So what we want to do is go through the process of generating an electrical signal, how that starts, how that stops, and what that means for the nerve cell. So if we look at a resting neuron and we look at the neuron cell membrane, what we find is that at rest it's said to be polarized. And what does that mean? That means that the outside of the cell is more positively charged than the inside of the cell. And the reason for that is that there's more sodium ions outside the cell than there are potassium ions inside the cell. You might ask, well, why is that? Well, that's for several reasons. Okay, one of the main reasons is that in every living cell in the body, there is a pump that uses ATP as an energy source and continually pumps 3 sodium ions out for every 2 potassium ions that it pumps in, and this counterbalances the leaking of sodium and potassium across the nerve cell membrane and retains this separation of charge when the neuron is at rest. So the inside of the cell is always negative relative to the outside when the cell is resting. What happens next is that we're going to generate now a change in this electrical state. And this is called depolarization, where a stimulus, now what's a stimulus? It could be a chemical stimulus, such as binding to a neurotransmitter and opening a door that lets charged particles move from one side of the nerve cell membrane to the other. It could be a physical stimulus that distorts a modified nerve ending that opens a gate physically that lets in charged particles, okay? Or it could be responsive to stimuli such as sound or to light or to any of a number of different physical stimuli, pressure for instance, or pain. All have the potential, depending on the receptor we're talking about, to open a door and to let a charged particle move from one side of the cell membrane to the other. So this stimulus, whatever it is, will depolarize the neuron's membrane. And by depolarize, what we mean is that we let positive charge flow into the cell through an open door. And in the case of the nerve cell, that positive charge is sodium. The membrane is now permeable to sodium because we've opened this gate, and a depolarized membrane will allow sodium to flow inside it and to change the charge. Okay, so what's happening now is that the separation of charge between the inside and the outside of the cell is becoming less and less. Okay, so the stimulus is doing this. What'll happen if enough of these tiny stimuli are received by the neuron is that they can eventually kind of run into each other and trigger at the beginning of the axon something called an action potential. Okay, the action potential is the movement of ions in an irreversible fashion that moves from the beginning of the axon all the way down to the end of the axon. And it doesn't change in magnitude, it doesn't change in speed. It's sort of like when you play dominoes and you flip over that first domino, all the rest are going to fall. The same is true when we're talking about an action potential. An action potential is an unstoppable force. Once we reach a certain critical voltage at a certain location in the neuron, the action potential is going to move from one end of the cell to the other. A graded potential exists where the inside of the membrane is more positive and the outside is less positive. And graded potentials can lead to action potentials, but only if they reach what we call the threshold voltage. So there has to be enough of a change due to stimulus from the graded potential, and it has to reach the beginning of the axon in order for the action potential to happen. Okay, so graded potentials and action potentials are related to each other, but if your graded potentials don't make it to the threshold voltage by the time they reach the beginning of the axon potential won't happen. The propagation of the action potential occurs if we reach a certain critical voltage at the beginning of the axon. This results from sodium moving into the cell and causes a wave of depolarization to move from the beginning of the axon all the way down to the end. The impulses will travel faster when the fibers have a myelin sheath around them because they will hop from node to node to node. Those nodes, remember, are the physical gaps between the Schwann cells in the peripheral nervous system and between the oligodendrocytes in the central nervous system. Okay, so that's a very rapid type of conduction, whereas if you're unmyelinated, your conduction is considerably slower. Repolarization occurs after this wave of depolarization has passed by a particular region of the cell. What happens is that potassium ions rush out of the neuron after the sodium ions rush in, and this ends up repolarizing the membrane. The repolarization involves restoring the inside of the membrane to a negative charge and the outer surface to a positive charge. And the reason that this happens is because once this voltage change passes this point in the cell, what will happen is that we will close the gates that let the sodium in, will open the gates that let the potassium out. And as the potassium continues to flow out of the cell, what will happen is that the voltage will dip back down actually below what we call the resting membrane potential. And then as those pumps, which are always working, continue to pump sodium out of the cell and potassium into the cell, eventually what will happen is that the resting membrane potential will be restored. And what will happen next, once that resting membrane potential is restored, is that that part of the neuron is now ready for the next electrical event. So if we look at repolarization, initial ionic conditions are restored because of the action of the sodium-potassium pump, which uses ATP and eventually restores now what we call the resting membrane potential. 3 sodium ions are ejected from the cell while 2 potassium ions are returned to the cell, and after a very short period of time, what happens now is that the resting membrane potential is restored. Now, what happens at the synapse? When the action potential reaches the axon terminal, the electrical charge opens calcium channels. The calcium will then rush into the nerve ending, into the ending of the axon, and that will trigger the fusion of these synaptic vesicles with the presynaptic membrane, and that causes a neurotransmitter to be released into the cleft. Now, this gap can exist between two nerve cells. It can exist between a nerve cell and a muscle cell, or a nerve cell and some other type of tissue, such as a gland, and it'll produce a physical response in the target. Calcium, in turn, causes tiny vesicles containing neurotransmitter to fuse with the axonal membrane. The entry of calcium into the axon terminal causes pore-like openings to form, and this releases the transmitter. The neurotransmitter molecules diffuse across the synapse, and they bind to receptors on the postsynaptic membrane of the next neuron, or whatever the next cell is, and that causes gates to open up that start a change in voltage on the target. So here you can see the calcium is already rushed in. The neurotransmitter-filled vesicles have fused with the presynaptic membrane, and now we are releasing neurotransmitter into the synaptic cleft. This is going to bind to receptors on the postsynaptic membrane. This causes ion channels to open up. This causes sodium to rush into the cell, depending on, again, the nature of the neurotransmitter and the type of receptor that it binds to. And this will cause a change in the voltage in the target. If enough neurotransmitter is released, graded potentials will be generated on the target. Eventually, an action potential can occur if sufficient graded potentials happen on that cell's membrane beyond the synapse. Okay, so you can see here the neurotransmitter binding to the gate and opening the gate up, and you can see the sodium rushing from the outside of the cell through this gate to the inside of the cell across the cell membrane, and causing the voltage to change in this tiny little region. The electrical changes prompted by neurotransmitter binding are very short. The neurotransmitter is quickly removed from the synapse, and again, the pumps that are there in the membrane will work to restore the original voltage situation and prepare the membrane now for the next electrical signal. So what I want you guys to watch now is a little video that talks about this in a little more detail. I think it's nice to see the process happen as opposed to talking about it step by step. There's a little bit more detail in this video than you need to know, but again, it's very useful in terms of what a graded potential is, what an action potential is, and what happens at the synapse between two nerve cells. So take a listen.
The human brain alone contains about 100 billion nerve cells, called neurons. A neuron, like every other cell, has positively and negatively charged ions, inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of -70 millivolts. The resting membrane potential depends on two factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allows sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at -90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to -70 millivolts. Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential. Neurons send signals over long distances by generating and propagating action potentials. Most action potentials originate near the axon hillock of the cell body, in the initial segment of the axon. It then travels the entire length of the axon. A closer look reveals that during an action potential, voltage-gated channels open and close, altering the permeability of the plasma membrane to sodium and potassium ions. A threshold stimulus changes the shape of the voltage-gated sodium channels, causing their activation gates to open. This event marks the beginning of phase 1 of the action potential, known as depolarization. As sodium ions diffuse into the axon, the membrane potential becomes less negative. This causes more voltage-gated sodium channels to open, and the membrane potential soars to +30 millivolts. At this point, two key events occur. The inactivation gates of voltage-gated sodium channels close, and voltage-gated potassium channels open. These two events mark the beginning of phase 2 of the action potential, known as repolarization. As potassium ions diffuse out of the axon, the membrane potential becomes negative again. However, the membrane potential continues in the negative direction, going beyond the resting state of -70 millivolts. This marks the beginning of phase 3 of the action potential, known as hyperpolarization. During this phase, voltage-gated potassium channels close and all voltage-gated sodium channels are released from inactivation. By the end of this phase, ions move through leak channels only and the membrane potential returns to the resting state of -70 millivolts. The neuron is now ready to fire another action potential. Summary: generation of an action potential. A threshold stimulus opens voltage-gated sodium channels. Sodium ions diffuse into the axon, depolarizing it to +30 millivolts. Voltage-gated sodium channels close, and voltage-gated potassium channels open. Potassium ions diffuse out of the axon, repolarizing it to a negative value. The membrane potential briefly hyperpolarizes. Voltage-gated potassium channels close, and the membrane returns to the resting state of -70 millivolts. Action potentials propagate in a continuous fashion in unmyelinated axons. Once an action potential is generated in the initial segment of the axon, it propagates the entire length of the axon. Recall, that a threshold stimulus causes voltage-gated sodium channels to open. The influx of sodium ions generates an action potential. It also establishes a depolarizing current that flows to the next segment and brings it to threshold. Voltage-gated sodium channels open, regenerating the action potential in this segment of the axon. Current flows from this segment and depolarizes the next segment to threshold, thus regenerating the action potential yet again. In this way, regeneration continues in one direction all the way down to the axon terminals. The basis for unidirectional propagation is revealed when we take a closer look. By the end of the depolarization phase of the action potential, all voltage-gated sodium channels inactivate and voltage-gated potassium channels open. These two events render this segment of the axon temporarily insensitive or refractory to another depolarizing stimulus. However, voltage-gated sodium channels in the downstream segment are closed and receptive to a depolarizing stimulus. Thus, propagation occurs sequentially down the axon to the axon terminals. In myelinated axons, action potential propagation is a bit different. Here they propagate in a saltatory or leaping fashion. The myelin sheath consists of multiple layers of tightly wrapped glial cell membrane. But this sheath is not a continuous one. Exposed areas of axonal membrane, known as nodes of Ranvier, occur at discrete intervals. Voltage-gated sodium channels are abundant in the nodes, but largely absent between nodes. So, action potentials are regenerated at each node, not in areas covered by the myelin sheath. However, the myelin sheath does provide the insulation necessary for the rapid spread of depolarizing current. And the sooner the nodes reach threshold, the faster action potentials propagate along the axon. Saltatory conduction is extremely fast. Velocities often exceed 100 meters per second. In contrast, continuous conduction is fairly slow. Velocities rarely exceed 2 meters per second. Nevertheless, both continuous and saltatory conduction propagate action potentials over varying distances because action potentials regenerate along the way. Summary: propagation of an action potential. Once generated, the action potential propagates the entire length of the axon without decrement.
Neurons communicate across junctions called synapses. When an action potential arrives at the end of an axon, a signal is transmitted across a synapse to a receiving cell. Although synapses may be either chemical or electrical, we will only look at a chemical synapse in detail. How do signals travel across a chemical synapse?
There is a narrow gap, the synaptic cleft, between the synaptic terminal of a sending neuron and the surface of a receiving cell. An action potential arrives at the end of the axon of the sending cell. This causes chemical changes that make vesicles containing a chemical called a neurotransmitter fuse with the plasma membrane. The neurotransmitter molecules spill out into the synaptic cleft, diffuse across, and bind to the receptors on the receiving cell's membrane. The binding of neurotransmitters to receptors causes the attached ion channels to open. Depending on the type of neurotransmitter, receptors, and channels involved, this may excite or inhibit the receiving cell. Here we show excitation. Sodium ions rush in, changing the polarity of the membrane, and an action potential is triggered in the receiving cell. The neurotransmitter is broken down by an enzyme, and the ion channels close. Enzymatic breakdown is one of the several mechanisms by which transmission is terminated at chemical synapses.
Okay, one of the easiest nerve pathways to understand is the reflex arc. This is a rapid, predictable, involuntary response to a stimulus that occurs over an extremely short pathway called a reflex arc, which essentially runs a direct route from a sensory neuron to an interneuron and straight back to an effector. Somatic reflexes are examples of responses that are governed by a reflex arc. They stimulate skeletal muscle, for example, the hot stove reflex, where you put your hand on a hot object and yank it away without actually having to process that information in your brain, is governed by a reflex arc. Autonomic reflexes regulate the activity of smooth muscle, the heart, and the glands. As an example, the regulation of blood pressure, digestive system function, and the heart rate are all examples of autonomic reflexes. So let's break down the elements of a reflex arc. There's 5 of them. The sensory receptor reacts to the physical stimulus to generate an electrical signal. The sensory neuron carries the electrical signal to the integration center, which is in the central nervous system. Specifically, it's in the gray matter of the spinal cord. This processes the information and then it generates a motor output. The motor neuron carries the message away from the central nervous system to the effector. The effector in this case, if you're talking about a typical reflex arc, say like the motor response to a hot stove stimulus, is going to be skeletal muscle. So this is designed to keep us alive. These are stereotypical responses that, if we had to think about them, would end up producing more damage than if we were to simply produce these actions as a result of a reflex. As an example, if you were to rest your hand on a hot object and then process the fact that you had done that and then assess that this was a damaging phenomena and then pull your hand away, by the time you've done all that, the damage is going to be much more extensive than it would otherwise be. So this is a sort of a rapid action that enables us to stay alive a lot longer than if we didn't have these reflex responses. And here you can see an example of a typical reflex arc where we have a physical stimulus, in this case pain. You can see a pin sticking here into the skin, stimulating pain receptors, and that's going to send an afferent signal from the sensory receptor into the back of the spinal cord through the dorsal root. The sensory neuron will then synapse on an interneuron that's contained inside the gray matter of the spinal cord, and then that will produce an electrical output directly to a motor neuron, which will send an electrical signal to the muscles in the affected area, and that will cause us to pull away from the painful stimulus. Now, the brain does get information about the painful stimulus, but it actually happens after the reflex has already executed. So the brain finds out about this really on the tail end of the phenomena. So again, you can see how these responses are designed to keep us alive. We don't have to think about them in order for them to happen. They simply occur. Now, some reflex arcs are faster than others. This is an example of a 2-neuron reflex arc. This is one of the simplest types. An example is the knee-jerk or patellar tendon reflex, where when you go to the doctor, he'll tap on the patellar tendon, which is a piece of connective tissue that runs from your kneecap to a bump on the front of the tibia called the tibial tuberosity. When he hits that tendon, what will happen is it'll produce pressure in that tendon, and that will yank on the patellar tendon. That response will be felt inside the spindle apparatus and the Golgi tendon organs that are inside the muscle, and that will send a sensory input into the central nervous system through the dorsal root, and that neuron will synapse directly onto a motor neuron without needing an interneuron, and the motor neuron will then immediately innervate, in this case, the quadriceps muscles to contract, and also the hamstring muscles to relax, and the leg will go up. So you'll engage in knee extension, and this doesn't require it doesn't require the brain in order to operate. The brain does become aware of it after the fact, but the stereotype response is basically just the result of these two neurons talking to each other. Because there's only one synapse in this nerve pathway, it's very, very quick. Okay, we just go directly from the afferent neuron onto the efferent neuron, and then from there out to the effector, and then we perform the activity. Don't have to think about it, it just happens. Notice again that the signal transmission is occurring in the white matter. You can see the white matter of the spinal cord involved, as well as the myelinated axons of the afferent and efferent motor divisions here, and the signal processing is happening primarily here in the gray matter, which is in the core of the spinal cord, this butterfly-shaped region right here. Now, some reflex arcs are a little bit more complicated. This is a 3-neuron reflex arc. It's made up of 5 elements: a receptor, a sensory neuron, an interneuron, a motor neuron, and an effector. And here you can see an example response to a painful stimulus. Okay, so here we have response to heat. You pull your hand away from the painful stimulus. As a result of the action of the arc, what happens is that the pain signal, the physical stimulus produced by the pain, hits the receptor. The receptor turns that into an electrical signal in the sensory neuron. That electrical signal runs all the way back through the dorsal root. It hits the gray matter. Here, an interneuron will take that information and process it, and then will synapse onto a motor neuron, which will carry electrical information away from the central nervous system and innervate the skeletal muscles necessary to withdraw from the stimulus. The effector in this case are the muscles of the arm. And so you can see here 1, 2, 3, 4, and 5 elements in the reflex arc. But what's accomplished is the same thing. We remove ourselves from the painful stimulus. As a result, we avoid damage that would have otherwise happened had we not had this ability to respond reflexively to this pain. Next thing we want to talk about is the central nervous system. And first thing we'll do is talk about where the central nervous system comes from and how the different parts of it develop. So when initially we're conceived, what happens in the female reproductive tract is that the sperm will fuse with the egg in a process called fertilization. At that point, what will happen is that the sperm and egg nucleus, the egg nucleus containing genetic material from the mother and the sperm nucleus containing genetic information from the father, will fuse, and that'll create a nucleus that contains 46 chromosomes, and that set of 46 chromosomes will be unique to that new individual. They won't exist in that same distribution in either the mother or the father. And so you're basically a composite of genetic material from your mother and your father. In addition to that, some of that genetic material in the sperm and the egg have been rearranged in the course of producing the sperm and the egg, so you are truly a unique individual. This is one of the great advantages of sexual reproduction, is that it produces diversity in the species. And as a result, if the species encounters environmental conditions that might make certain members of the population sick or may kill them, not all of those members of the population will die because of the genetic differences that produce physical variety in the population, what we call diversity. Once the sperm and the egg nucleus fuse, the cell will begin a rapid series of divisions that'll result in a cluster of cells that's called a morula. And those cells are going to have all the same genetic material in each and every cell, but that genetic material is going to be unique to this new individual. As that morula continues to divide, eventually what will happen is it'll change from a solid ball of cells into a hollow ball of cells that's called a blastocyst. And the blastocyst will then invaginate and form a little groove on the top, which is called the neural tube. So let's draw this process here. So this is the zygote, the sperm meeting the egg for the first time. That'll be our little cell. And then what happens is we proceed along and we become now a cluster of cells. This would be the morula. Okay. And then what will happen is that the cells will kind of move around a little bit and form a ball of cells called the blastocyst. So we're just going to take a cross section through this ball of cells. The inside of the ball is filled with fluid, and the perimeter of the ball is basically a sheet of cells. And then what will happen is that this hollow ball will indent on one side and form what we call the primitive streak. So we're still looking at basically a sagittal section, transverse section through this now a disc of cells. So the ball has indented on one side and formed a groove at the top. And you might ask, well, how is all this happening? Well, this is all happening because there are instructions that are being produced by the cells in the form of tiny chemicals that get extruded into the extracellular environment, and they serve as chemical messages to other cells to tell them where to go and what to do when they get there. Okay, so these control things like what the cell is going to look like, where the cell is going to move to, and once the cell gets to that location, how that cell is going to change how it looks and acts. So this is the blastocyst, and then this is what we call the embryonic disc. The next thing that's going to happen is that cells that are here on the top of this disc are going to migrate through this little groove and are going to start to populate now the inside of this hollow space that's filled with fluid. And at that point we've done something pretty significant, which is that we have formed now what we call the three primary germ layers. Okay, so let's draw now the situation. Okay, and then inside here we've got little cells here that came from this upper layer and are now inside this hollow chamber. And this marks a significant point in development called the establishment of the three primary germ layers. Okay, and you might say, well, what are the three primary germ layers? Well, they're the ectoderm, which is this area here on the top. Okay, draw a circle around it. This is the ectoderm. The mesoderm, which are these cells that are here in the middle, and then the endoderm, which are these cells on the underside. And from those populations of cells, all the other structures of the embryo will form. The ectoderm will produce the skin and the nervous system. The mesoderm will produce the cardiopulmonary and musculoskeletal system, and the endoderm will produce a lot of the internal organs. So what happens next is that this little groove that was on the top of the disc will eventually drop down below the ectoderm and form a little tube called the neural tube. Okay, so now this represents the tube dropping down below. So this thing has basically pinched off and dropped below the level of the ectoderm. And this is eventually going to be your spinal cord. It isn't yet, but it will be soon. Remember that we're looking at a transverse section, so you could imagine in terms of the embryo that the front of the embryo is coming out of the slide at you, and the legs of the embryo would be poking into the slide away from you. So this is a transverse section. Okay, so it's at this point that we pick up the rest of the development of the nervous system. So the central nervous system develops from the embryonic neural tube, which is that structure that dropped below the ectoderm immediately after the formation of the three primary germ layers. The neural tube will become the brain and the spinal cord. The brain will be up towards the head, and of course the spinal cord will run most of the region of the thoracic and the lumbar region. Won't reach all the way to the end of the lumbar region as the adult. What'll happen is it'll be a little bit short of running all the way to the sacrum, and so what happens below the region of the upper lumbar is that a whole bunch of nerves will actually radiate out from the lumbar spine, and we'll see that in just a bit. The opening of the neural tube becomes the ventricles, which are hollow chambers that are inside the brain and the spinal cord that are filled with cerebrospinal fluid, which helps to maintain a relatively happy and healthy environment for the spinal cord and the brain. You could think of it as the extracellular environment for the neural and the glial cells. There's 4 ventricles inside the brain, 2 lateral ventricles, a third ventricle that's in between them that are connected to the lateral ventricles by tiny openings, and then there is a cerebral aqueduct that connects the third ventricle to the fourth ventricle, and then the fourth ventricle is connected to the central canal, which runs right through the middle of the gray matter of the spinal cord. And in the third and the fourth ventricle are openings. The openings in the third ventricle connect it to the lateral ventricles and to the cerebral aqueduct, and the opening in the fourth ventricle allows fluid to move down the central canal and also to move out of the fourth ventricle and bathe the entire outside of the brain and the spinal cord. And we'll see that in just a moment. So those are the 4 ventricles in the brain, and then of course the central canal and the spinal cord. Now you might say, okay, well, what happens once that, first of all, where does that cerebrospinal fluid come from? And the answer is pretty straightforward. The cerebrospinal fluid is basically filtrate of the blood, and so it's a lot of the elements that you find in plasma, with the exception of some of the larger plasma proteins and the formed elements. There's no, for instance, red blood cells or white blood cells or platelets in cerebrospinal fluid. They stay in the bloodstream, and there's not a lot of large plasma proteins in the cerebrospinal fluid. That blood filtrate bathes the brain and it bathes the spinal cord, and then it'll eventually return to the circulatory system through little protrusions that are located in a layer of membrane that surrounds the brain called the arachnoid mater, and it's through this that the fluid will diffuse eventually back into the bloodstream and rejoin the circulation. So that's the one-way flow of cerebrospinal fluid around the nervous tissue. And again, the reason that it's important is because it maintains the temperature around the brain and the spinal cord. It helps to maintain pH and maintain electrolyte levels, and it's a way to remove waste products. And it's also the medium inside which the brain and the spinal cord float, and that protects them from damage so that they don't bump against the bones that surround them, which would produce damage. Okay, so you can see here what's happening at 13 weeks. Already you can see the beginnings of what's going to be the cerebral hemispheres, which you think of as the brain proper. And then inside, what you've got is the brain stem, which is made up of the medulla oblongata, the pons, and the midbrain, and we'll see those in just a minute. And the midbrain attaches the cerebral cortex and the deeper cortical matter to the brainstem. The brainstem connects all that to the spinal cord. So you can see how these parts of the neural tube are beginning to change. And so here you can see the adult brain, which is made up of the cerebral hemispheres, the diencephalon, the brainstem, and the cerebellum. Okay, so you can see each of these parts. There is the brainstem there in green. Okay, there's the diencephalon right here, and these are the cerebral hemispheres with the white matter underneath it. So let's start with the cerebral hemispheres. This is the part of the brain that you probably think of most often when you think of the brain. It has paired superior parts called hemispheres, one on the left and one on the right. This includes more than half of the brain mass. The surface is made up of ridges called gyri and grooves that are called sulci. And here you can see again the cerebral cortex. So there are several lobes in the cerebral cortex: the frontal lobe, the parietal, the temporal, and the occipital lobe. Down here you can see the cerebellum, and here of course is the brainstem. You can see the medulla oblongata here and the pons. The midbrain and the thalamus, which are part of the diencephalon, are covered up by all this cerebral cortex. And what you want to notice about the brain is the fact that it does have a lot of grooves and ridges in it, and the reason for this is to increase the amount of surface area in the brain that's available for electrical work. Okay, so anytime you see a whole lot of wrinkles and grooves and ridges in an organ, it's usually because we need to increase the amount of available space to do whatever job it is that we have before us. Now, if you look at a cross section through the surface of the cerebral cortex, what you see is that the gray matter, which are primarily the cell bodies, are on the top, and there's white matter underneath it. So again, the signal processing is happening on the surface of the cerebral cortex, and the signal transmission is happening in the deeper areas of the brain. In fact, when the brain develops, one of the things that happens is that the nerve cells actually are born deep inside the brain, and then they crawl towards the surface of the brain, and when they get to the surface they stop, and then they change into functioning neurons, and then newer neurons will actually climb over the top of them and continue the process of brain development. So the youngest part of your brain is actually on the surface, and the older parts are deeper down inside. So it's the opposite of the way that tree rings are, where the newest part is on the inside and the oldest is on the outside. Now, if we look at the different lobes of the cerebrum, we have several surface lobes that are easily recognized as a result of the grooves that separate them. These include the frontal lobe, the parietal, the occipital, and the temporal lobe. Now you can see here the temporal lobe. This is the frontal lobe here. Back here is the parietal lobe. This is the occipital lobe. And then there's a fifth lobe of the brain that's deep down between the temporal and the parietal lobes. It's called the insula, and to see that we'd have to move these two lobes out of the way. Now, what do each of these parts of the brain do? What are some of their broad job descriptions? Well, the frontal lobe is the site of our higher order thinking. This is where our reasoning and analytical power is located. You might say that this is also where the personality is housed. It also happens to be the place where we plan motor activities and then send them to a ridge of tissue that's called the precentral gyrus, which you can see running right along the groove of the brain here. Okay, and this is where the finalized motor commands will be sent down to the deeper areas of the brain, then to the brainstem, and from there to the spinal cord, and from the spinal cord to the peripheral nerves, where the muscles will then carry out the activity. The parietal lobe is the home of what we call the sensory association area. So this is where information from all over the body and inside the body comes. It enters the spinal cord, runs up to the brainstem, and from the brainstem it runs to a ridge of tissue called the postcentral gyrus, which you can see here. Okay, and what it does is as it receives sensory information, will send it to the sensory association area, which is in this part of the parietal lobe, which will then assign categories to the information. Is this light? Is this sound? Is this temperature? And so on. And then that information will be integrated in other parts of the brain, and then the frontal lobe will decide what to do about that sensory input. The temporal lobe is the area of the brain that receives information about sound and speech, and also about our sense of equilibrium, the ability basically to stand up straight and not fall over when we move. The occipital lobe of the brain is the area where we receive information about sight. This is where our primary visual cortex is located, and so the second cranial nerve, the optic nerve, projects back here to the occipital lobe, and this is part of one of several areas in the brain that are able to turn information that comes to us in the form of light and color and turn it into what we perceive as images. So the primary somatic sensory area of the brain receives impulses from the body's sensory receptors. It's located in the parietal lobe in an area called the postcentral gyrus. The primary motor area sends impulses to skeletal muscle. That's the precentral gyrus, and that's what we were talking about, where the finalized motor commands get sent to the brainstem and from there to the spinal cord. And it's located in the frontal lobe. Broca's area is involved in our ability to speak, and you can see these different areas of the brain right here. So here you can see this is the frontal lobe, all right, and it's broken up into the motor association areas. This is the premotor area, and this is the primary motor cortex. So information comes into the sensory area of the brain through the brainstem up to the postcentral gyrus. The postcentral gyrus, as it receives the information in the primary somatic sensory area, will assign categories to that sensory information. Okay, is this sound, is this light, is this heat, and so on. And then it'll send it from the sensory association area to other areas of the brain. For instance, it'll send it to the frontal lobe, and it'll send it from there to the premotor area if we want to do something about this sensory information, and then the finalized motor commands will be sent to the primary motor area and will carry out some sort of activity. The primary somatic sensory area and the primary motor area are divided from each other by the central sulcus, this groove that runs right between these two ridges of material. And this marks the dividing line between the parietal lobe and the frontal lobe, but it also marks the dividing line between the primary motor cortex and the primary somatic sensory area of the brain. You can see other areas of the brain that are important in normal function. The olfactory area of the brain is basically underneath the parietal lobe. This is where we process information about odors. Broca's area are the motor responses that are engaged in the production of speech. Language and comprehension here in the frontal lobe. Working memory and judgment also in the frontal lobe, as well as problem solving. And of course, we've already talked about the association area. Back here you can see the visual cortex, you can see the auditory area in the temporal lobe. You can also see the area of the sensory association area that's dedicated to the comprehension of speech and language. This is part of what goes into our ability to communicate. Now, if we were to look at these two ridges of tissue, the primary motor cortex and the primary somatic sensory area, what we would find is that different parts of the body either input or are output to by different regions of these tissues. Okay, so this, for instance, would be the primary motor cortex map. And what these different areas of the cortex represent are different parts of the body that are governed by information that comes from these different parts of the cortex. So, for instance, a lot of the motor commands that control facial movement are located on the lateral portion of the motor cortex. Here you can see, for instance, the lips, the jaw, the tongue, the throat, the eyes, the neck, and so on, located in this region. You can see that the upper extremities are primarily towards the superior lateral portion of the motor cortex, and then the lower extremities and the trunk are down into the groove that separates the two hemispheres from each other in this part of the cortex. But the other thing that I want you to notice is the fact that these parts of the brain aren't equally divided. The different amounts of surface area dedicated to each part of the body, and the reason for that is that different parts of the body require different levels of motor control. So this is why, for instance, the hands, the face, the tongue are overrepresented in the motor cortex, while the trunk and the legs are underrepresented. With the sensory cortex, it's essentially the same, but with some slight differences. For instance, you've got more sensory information coming in from the feet, so they are overrepresented in the sensory cortex. You can see sensory information, for instance, coming from the genitals. Again, notice that the face, the lips, the nose, the eyes, and the hands, as well as the teeth, tongues, jaw, and tongue, and pharynx are overrepresented in the sensory cortex. And again, that's because there are more sensory receptors in those parts of the body than there are, say, in the trunk or the arm or in the leg. Okay, so that's the explanation for why this map is the way it is. And you might well ask, well, how do we know these things? And The way that we know these things comes from two sources of information. One, we can directly stimulate these areas of the brain electrically and have the patients report sensation in different parts of the body. We can also stimulate, say, the motor cortex and produce motor responses in those parts of the body. And then in the sensory region, we can stimulate different parts of the body electrically, and the patients can report sensation in those regions. The other line of evidence that comes not only about this part of the brain, but from all the parts of the brain, comes from individuals who had brain damage to specific regions, and the physician, the clinician, will record deficiencies in certain brain function in people who've had certain types of damage, and then will impute that those parts of the brain are responsible for those functions. So cerebral areas that are involved in special sense include the gustatory area, which is our sense of taste, our visual and auditory area, which is sight and sound, our olfactory area, which is odor, and then interpretation is found in the cerebrum, where we have a speech and language region, a language comprehension region, and also a general interpretation area. And again, this is just showing you the different parts of the brain that are dedicated to these different functions. So what I would do is I would star this figure in your textbook and make a note of where these regions of the brain are, because I can guarantee you that you'll be asked on a quiz or an exam, if a certain part of the brain was damaged, what functions might you expect not to work the way that they should? And if you know this map of the brain, then those questions will be relatively easy to answer. Okay, if we look at the cerebrum in a little bit more detail, we see that it has several layers. The gray matter is an outer layer in the cerebral cortex that's made of neuron cell bodies, while the white matter are fiber tracts deep to the gray matter that are involved in signal transmission. The corpus callosum is a tract of white matter that connects the two hemispheres to each other, and the basal nuclei are islands of gray matter that are buried within the white matter that do some more signal processing before it gets sent onto the brain stem, which consists again of the pons, the medulla oblongata, and then from there it's going to be sent eventually down to the spinal cord. So here you can see the diencephalon, which sits on top of the brainstem, and it's enclosed by the cerebral hemisphere. It has three parts: the thalamus, which is called the gateway to the cerebral cortex, the hypothalamus, and the epithalamus. The hypothalamus lies underneath the thalamus. The epithalamus lies above the thalamus. Okay, so again, the word will tell you the location of these different parts of the brain. Essentially, what the thalamus does is it allows certain stimuli to get to our conscious awareness, and then it keeps other stimuli at the level of the brainstem, where the brainstem can still respond to those stimuli, but we won't be aware of that response on a moment-to-moment basis. The hypothalamus is in charge of the master gland of the endocrine system called the pituitary gland, and it does this by producing chemical signals that tell that gland to produce its chemical signals, which talk to the rest of the endocrine system and regulate its function. The hypothalamus also controls things like heart and respiratory rate and body temperature. It also has a role in hunger. A lot of different functions, primitive functions, are governed at the level of the hypothalamus, which again, of course, is part of your brain. Here you can see a look at a frontal section through the brain, and what we're showing you here are different nerve pathways that carry information to different locations in the brain. So let's start with something relatively straightforward. These sort of greenish lines here, greenish yellow lines, those are called commissural fibers. They're part of the corpus callosum, and this is how the right half of the brain knows what the left half of the brain is doing and vice versa. And if you think about that, that's a pretty important thing to coordinate, because if the right and left side of your body were operating independently of each other without paying attention to what the other side was doing, it would be very difficult for you to do things like walk across the room. Okay, you have to coordinate the action of one side with the other in order to carry out purposeful activity. Another easy set of tracks to understand are these, what we call projection fibers, and what they do is they carry information that comes from the cerebral cortex to the white matter, and they carry it down to the brain stem, to the cerebellum, and ultimately from there down to the spinal cord, which will carry the information to the peripheral nerves, which will eventually reach the effectors. In the case of the somatic motor system, those are our skeletal muscle, and in the case of the autonomic nervous system, there are things like our smooth muscle and our glandular secretions and the activity of the heart, as well as the digestive system, just to name a few. Now, the other fibers are called association fibers, and what they allow to happen is for information that's received or transmitted from one part of one hemisphere of the brain to reach another part of the same hemisphere. So this is part of the way that information gets moved around inside the hemisphere of a brain. So you can imagine that that these tracks are kind of like a map, a highway map of the brain, and these are the routes that the information follows when it goes from one place to the next. So using a combination of these association fibers, you can see, for instance, how say a plan to do something gets generated in, say, the frontal lobe of the brain. It eventually gets sent back through these association fibers all the way to the precentral gyrus, the primary motor cortex, and then from there, that information would cross these commissural tracts to the other side of the brain, so that again the right and the left half of the body would be able to coordinate their activity, and then that finalized plan would be sent through the projection fibers, which would ultimately reach now the brain stem, the spinal cord, and from there the peripheral nerves. Okay, so that's essentially how these fibers are directing information. Other regions in the brainstem that are important to note include the reticular formation, which is a column of gray matter in the brainstem, which is involved in the control of visceral organs, our internal organ function. It's also involved in waking up the brain so that it's aware of incoming stimuli, and this is a system that uses the reticular formation to coordinate the stimulation of the cerebral cortex in what's called the reticular activating system, which plays an important role in wakefulness and in sleep. So here you can see the location of the reticular formation. Again, there's the medulla oblongata, the pons, the midbrain. Okay, there you can see the thalamus. All right, there's the hypothalamus. There's the epithalamus. There's the corpus callosum. Okay, the white matter that connects the right and the left half of the brains together. Okay, and this is the cerebral cortex up here. This is our conscious awareness. And so you can see with the reticular activating system, what happens is that incoming sensory information, say through the eyes and through the ears and so forth, what they do is they enter the reticular formation, because these are afferent pathways as well as our general senses, and then the reticular activating system will allow these sensations to pass through the thalamus and get to our conscious awareness. This is, for instance, how we become aware of our alarm clock ringing in the morning, or the smell of coffee or bacon for breakfast. Okay, the reticular activating system lets this stuff through, and we become slowly aware of it. If we have damage to the reticular activating system, that can result in things like coma, where we're unable now to respond to our external environment because the stimuli aren't being allowed to pass through the thalamus and ultimately get up to the cerebral cortex. You can also see that there are descending motor pathways that project down from the brainstem to the spinal cord. This is how we send commands from the cerebral cortex to the brainstem down to the spinal cord, and then the effectors carry out the activity. Okay, if we look under the section of the brain known as the diencephalon, one of the primary structures located there is the thalamus. It surrounds the third ventricle, which lies between the two lateral ventricles, and it's a relay station for sensory impulses. It serves as what we would think of as the gateway to the cerebral cortex. It transfers impulses to the correct part of the cortex for localization and interpretation. It also acts as a filter to allow certain stimuli to get to the conscious areas of the brain, the cortical areas, so that we can respond to them appropriately. So you could think of it as a filter. The hypothalamus is directly underneath the thalamus, and it's an important autonomic nervous system center structure. It regulates a lot of very important homeostatic functions such as body temperature, water balance, respiratory and heart rate. It also regulates metabolism. It houses the limbic center of the brain. Now, what is the limbic center or limbic system? You could think of that as our emotional brain. It's the part of our brain that provides a qualitative judgment on various stimuli as they come in. For instance, you might smell a flower and be aware of that smell, and you might think of that odor as pleasant. The part of you that says that that's a pleasant odor is coming from the limbic system. You might smell the odor of something spoiling in your refrigerator, and you might say that that's a bad or an unpleasant odor. And again, that value judgment would come from the limbic system. So our emotional brain. And where is the limbic system exactly? It sort of surrounds the corpus callosum and the three regions of the thalamus, the epithalamus, the thalamus, and the hypothalamus, and is made up of the cingulate gyrus, the olfactory bulb, the amygdala, and the hippocampus. And again, it serves as a way of applying emotional value judgments to stimuli. It also regulates the nearby pituitary gland. This is the hypothalamus, and it produces two hormones of its own. Those are oxytocin and vasopressin. And as we'll find out in the chapter on the endocrine system, oxytocin is important in regulating responses such as uterine contractions and the milk letdown reflex, and vasopressin is our body's hormone that helps regulate body fluid by allowing us to retain more of our body water and reducing urine output. And those hormones are actually manufactured in the hypothalamus. Okay, so the limbic system is the overall structure of the brain in which the hypothalamus is a part. Okay, hypothalamus is a part. Okay, the epithalamus is above the thalamus, and it forms the roof of the third ventricle and houses the pineal gland, another endocrine gland that secretes a hormone called melatonin, which is important in our sleep-wake cycles, what we call our circadian rhythm. Basically, our body's sense of when we should be active and when we should be inactive, and that's generally associated with the 12-hour periodicity of light levels. We're inactive when it's dark out, and we're active when it's light out. It includes the choroid plexus, which is where the cerebrospinal fluid is manufactured. The choroid plexus is the site where the blood is actually filtered, and a portion of that filtrate becomes the cerebrospinal fluid. And remember that the cerebrospinal fluid provides a nice, stable environment for the neurons and the glial cells to live in. It maintains a constant pH and temperature and electrolyte concentration around the neurons and the glial cells, and it also serves as a method of removing waste products when they accumulate outside the neurons and the glial cells. The brainstem attaches directly to the spinal cord. It has three parts: the midbrain, the pons, and the medulla oblongata. The midbrain is the portion that connects the thalamus to the brainstem. The pons connects the midbrain to the medulla, and the medulla connects the pons to the spinal cord. Okay, so this is the most superior structure in the brainstem, and this is the most inferior structure. So let's start with the midbrain. It's mainly composed of tracts of nerve fibers. So if you were to look inside the midbrain tissue, what you would find is a lot of white matter in there. And it has two bulging fiber tracts called the cerebral peduncles and four rounded protrusions on the back of the midbrain called the corpora quadrigemina, which contain centers that process information related to vision and hearing. The corpora quadrigemina, that's Latin for four bodies. These are four bulges on the back side of the midbrain. The upper two bulges are there to process visual information. The lower two bulges of these four bodies are there to process information related to sound and hearing. The pons is the bulging middle part of the brainstem, and it's made up of fiber tracts. So there's, again, a lot of white matter in the pons as well, and it includes nuclei that are involved in the control of breathing when we're unconscious. Now again, here when we say nuclei, we have a very specific meaning for the word nuclei. What we mean are special areas of tissue that have a certain dedicated task inside that organ. So when we say that there are nuclei in the pons responsible for breathing, we say that there are certain regions of neural tissue in the pons that govern those functions. And we find that when those areas of the brainstem don't work the way they're supposed to, sometimes when we're unconscious, breathing will stop, and this is a condition called central sleep apnea. What it results in is a lack of innervation of the diaphragm, which is the main muscle of breathing. And when the diaphragm isn't working, then we're not able to ventilate the lungs. And if we can't ventilate the lungs, then very soon respiration stops, and you could be in danger of imminent death if we don't restore the periodic innervation of the diaphragm as soon as possible. So it's thought in a lot of circles that failure of these nuclei is sometimes what contributes to a condition known as SIDS, or sudden infant death syndrome. We think that in a certain percentage of these infant deaths that it's a failure of the breathing centers in the pons to properly innervate the diaphragm when the infant is asleep that results in the cessation of breathing and then the infant's death. Now, we don't know this for certain because we would have to actually witness it while it happens and be monitoring the pons in order to assess that loss of function, but it's what we would call an educated guess. Now, the medulla oblongata is the lowest part of the brainstem. It merges directly into the spinal cord, and it includes several important fiber tracts. So again, there's a lot of white matter in the medulla oblongata. It contains control centers that govern heart rate, blood pressure, breathing, swallowing, and vomiting. So a lot of reflex centers are housed in the medulla oblongata. It's the most inferior part of the brainstem. It's what connects the pons to the spinal cord. Now, the cerebellum is located posterior to the brainstem, and it's made up of two hemispheres that have a convoluted surface similar to the cerebral cortex, but its texture on the surface looks a lot more like, if you can imagine this, turkey burger. And again, the reason for these ridges and grooves is to provide more surface area for the cerebellum to do its job, which is to coordinate what you want to do with what you are actually doing. So what the cerebellum does is it takes sensory input, it compares that to motor output, and it corrects actions on the fly if the intentions don't match up with the perceptions. So you could think of it as a motor action editor. It provides involuntary coordination of body movement, and you can see all of these different structures here in this sagittal section of the brainstem. So what we're looking at here are the three regions of the brainstem. There's the medulla oblongata, the pons, and the midbrain, and there's the thalamus and hypothalamus. And you can see up here this upper region, the epithalamus, okay, the part that is above the thalamus. There is the corpus callosum. Those are the commissural tracts that connect the right and left hemispheres of the brain. You can see, of course, the cerebral cortex through here. And I'm sure you're probably curious, where was that limbic system you were talking about earlier? And if I were to trace it, it would basically run from here around to here, and then there's a structure called the hippocampus that would run right in front of this, and then the olfactory bulbs, which you can't see in this particular picture. So it basically rings the third ventricle and the corpus callosum. You can see back here the pineal gland, which produces melatonin that controls our sleep-wake cycles. There's the corpora quadrigemina, which receive and process visual information in the case of this upper pair of bulges on the back of the midbrain, and auditory information in the case of this lower set of bulges. There is the cerebellum, the basically on-the-fly editor of our intentional movement to make sure that it matches up with our perception. And then if we were to look for the ventricles of the brain that would be near this region, the third ventricle would basically surround the thalamus. Okay, this actual piece called the intermediate mass is what connects the two halves of the thalamus. If you were to look at the brainstem and the diencephalon, if you were to remove them from the cerebral cortex and the overlying white matter, what you would find is a structure that looks a little bit straight on, head on, like a double-headed mushroom. Okay, so where this would be the stalk of the mushroom and the two thalami would be the two mushroom caps. Okay, that's basically what it appears like. So this would be the third ventricle lying between the two thalami, and then down here, this is the fourth ventricle. This is the cerebral aqueduct. This is how the cerebral spinal fluid flows from the third ventricle to the fourth ventricle. And then the fourth ventricle is continuous with this central canal that goes right down the middle of the gray matter of the spinal cord, but it also has median and lateral apertures that allow the cerebrospinal fluid to escape and bathe the entire outside of the brain and keep it in a relatively stable, steady environment. You can also see, looking at the cerebellum, that there's white matter in the core and gray matter on the surface, just like there is in the cerebral hemispheres. There's gray matter on the surface and white matter deeper down. So the signal processing, again, happens on the superficial surface of the cerebellum, and the signal transmission is what's happening in this deeper, whiter matter that looks a little bit like a tree, and that's why it has the nickname the arbor vitae. Other structures that you can see the occipital lobe, the parietal lobe, the frontal lobe here. The temporal lobe you can't really see because we're looking basically at a sagittal section right through the middle of the brain, but the temporal lobe would be approximately here. Okay, and the other structures that we can pick up are the pituitary gland, which lies right under the hypothalamus, which is the gland that governs the rest of the endocrine system. And here you can see the optic chiasm. That's the place where the second cranial nerve crosses from left to right just under the floor of the brain. Chiasm means to cross. So this is where the an optic nerve that starts on the right half of the brain crosses over to the go into the back of the left eye, and vice versa. Okay, so again, keep this in mind when we talk about some of these other structures in the brain. Now, you can also see, if you look very carefully, these little red areas here. There's a red area in every ventricle of the brain, the two lateral ventricles, the third and the fourth ventricle, and those are the choroid plexuses, and that's where the cerebrospinal fluid is actually manufactured, and it flows through all these internal spaces inside the brain, and then also, as we noted earlier, flows all around the outside of the brain and the spinal cord, and also through the middle of the spinal cord, right through the central canal that pierces the middle of the gray matter. If we were to look behind this window, this is called the septum pellucidum, we would see the lateral ventricle. It lies just beyond this thin membrane. Okay, so that's to orient you. And if you're curious as to what the lateral ventricles look like, they look just like Cincinnati Reds logos. Okay, so if you're a baseball fan, you can look up the logo and see that the little lateral ventricle runs right through here in one half of the brain, and a similar one is on the other hemisphere inside the brain as well. Okay, so what I'd like you to do is watch this quick video that talks about how the brain functions, and I'll rejoin you in a discussion about the protection of the central nervous system. So stay tuned.
Your brain, the most complex organ in your body, the seat of who we are, what we are, the container for our thoughts and our memories. In short, we are our brains. Our brains are built from many parts that perform different functions. Deep in the centre of the brain is the brainstem. This is the oldest part of the brain and has also been called the reptilian brain. It controls many essential functions such as breathing and heart rate or blood pressure. It's whether we feel hungry or thirsty. It's also the seat of our most fundamental emotions: happiness or sadness, fear and love or hate. They all seem to reside in the brainstem. Sitting atop the brainstem is the thalamus. This is like an older version of our brain, which now acts as a gateway to the higher cortical regions. All sensory information from our bodies, whether it's sensations from the skin or a sense of touch, passes through the thalamus and is directed to the correct regions of the cortex for further processing. Surrounding the thalamus is a structure called the hippocampus. This is a crucial part of the brain. It's the seat of our spatial working memory. It lets us remember where we put those keys, or how to get back home from work or school. This region has even been shown to be bigger in London cabbies after doing the knowledge. The folded outer surface of the brain is called the cerebral cortex. Although it all looks the same, it's actually divided into different lobes that perform very different roles. The occipital lobe contains the visual cortex, and so is concerned mainly with vision. The parietal lobe is concerned with processing sensory information and integrating it with the visual information from the occipital lobe. The temporal lobe is the centre for memory and learning. It contains the hippocampus and a number of other regions that are required for us to recognise different objects, faces, sounds and environments. The frontal lobe is probably the most important part of the brain for defining us as who we are. It's where our higher emotions and personality reside, as well as language and social behaviour, lots of the things that we think of as being human. It's our decision-making centre and contains the motor cortex, which controls voluntary movement. At the back of the brain are the two lobes of the cerebellum. This region of the brain is a coordinating centre for movement, allowing us to move multiple sets of muscles in a coordinated fashion so that we can walk and talk, pick things up and put things down. So what are our brains made of? Muscle or bone? Well, there's none of that in the brain, even though it takes over a fifth of the calories you eat just to keep it running. Instead, your brain is made up of billions upon billions of cells called neurons. These cells are very specialised, with long extensions through which they talk to each other and form large and complex networks. These networks are the basis of how the brain does what it does. The processes are of two basic types. Dendrites receive incoming information from other neurons. Signals pass through the cell body or soma, where, if sufficient information is received, a signal is passed to the other neurons via the axon. This is happening in billions of neurons across the brain 24 hours a day, 7 days a week. It's happening right now while you're watching this. So how do neurons talk to each other? Neurons are usually seen to use electrical signals to communicate. However, the act of passing the signal from one neuron to another is actually a chemical process. This happens at structures called synapses. These are formed from the terminal of an axon and a swelling on the dendrite called a spine. Electrical signals from the axon pass to the dendrite via a chemical messenger called a neurotransmitter. Neurotransmitter is stored in the small vesicles inside the axon terminal, or the presynaptic bouton. When a signal arrives down the axon, one or more of these vesicles joins with the outer membrane of the bouton and releases its contents into a small gap between the axon and dendrite. This is the synaptic cleft. On the other side of the synaptic cleft are receptor molecules to which the transmitter binds in much the same way as a key fits into a lock. The receptor molecules act as pores through the outer membrane of the dendritic spine, allowing charged atoms or ions to cross. This entry of charged ions into the spine recreates the signal that was sent from the previous neuron, and in this way information can be passed from one neuron to the next and around the neuronal network. Although we've seen only one neuron here talking to one other neuron, in reality each neuron talks to many others and can have thousands upon thousands of spines. In this way, an extraordinarily complex system can be built that allows us to see, hear, to experience the world around us. It allows us to walk and talk, and to laugh and cry, and to be who we are. So we really are our brain.
Okay. The central nervous system is so important that it has to be well protected from damage, because the neurons in the central nervous system do not undergo cell division and they do not repair themselves to any great extent. And as a result, since you've got all the neurons you're going to get, and the only thing that happens as you age is that they die, we need to protect them as much as we possibly can. So what are some of the protective layers in the nervous system? Well, this includes the scalp and the skin, the skull and the vertebral column, which the skull protects the brain and part of the brain stem, and the vertebral column protects the rest of the brain stem and the spinal cord. And then the meninges, which is a series of 3 membranes that surround the brain and the spinal cord. They're made up of the arachnoid mater, the pia mater, and the dura mater. And then the cerebrospinal fluid, which is that filtrate of blood that's generated in the choroid plexuses of all 4 ventricles, and then flows both within those open spaces inside the brain as well as around the outer surface of the brain and the spinal cord. And again, that serves to maintain a steady environment so that the neurons can do their job for as long as possible. The blood-brain barrier is generated as a result of ependymal cells that line the ventricles of the brain and the spinal cord, and what they do is they keep the formed elements of blood from coming into contact with nervous tissue per se. What do I mean by the formed elements of blood? I mean the red blood cells and the white blood cells. So they never actually touch nerve tissue or glial tissue. However, because the glial cells are very good at extracting nutrients from the circulation and oxygen, as well as disposing of waste products, it's not necessary for the blood, the formed elements of blood, to come into contact with the nerve tissue. As a result, the brain and the spinal cord enjoys something that very few other organs in the body enjoy, and that's a quality called immune privilege. Essentially, because they're isolated from the white blood cells, we don't have to worry about the phenomenon of tissue rejection if we were to require a transplant of tissue into any one of these immune privileged spaces. And what are the organs in the body that have immune privilege? Those include your brain stem, and your spinal cord, your liver, the corneas of your eyes, and for men, the lumen of the vas deferens, and for women, the lumen of the fallopian tubes. Those are all off limits to the white blood cells that would normally mediate the rejection response. And so here you can see the protective layers of the brain from the outside to the inside. So this is the skin of the scalp, and this is the periosteum, the nutritive membrane that supplies oxygen and nutrients and some protection to the flat bones of the skull. You can see underneath it the flat bones of the skull. Notice the layer of compact and then spongy and then compact bone again. And we talked about the fact that because of this arrangement of compact, spongy, and compact bone, any force that's applied to the flat bones of the skull will be distributed over a very wide area, and as a result, it's very difficult to apply enough force to the skull to actually penetrate this bone unless it's an extremely high amount of force, and that helps protect the brain from physical damage. Okay, so this flat bone, the flat bones that surround the cranium, okay, the parietal, the frontal, the temporal, and the occipital bones, they all have this feature, and as a result, the brain is protected from these kinds of shocks. Now, even underneath the flat bones of the skull are three layers of membranes that protect the brain: the dura mater, which is the outermost fibrous sheath; the arachnoid mater, which is so called because it has web-like extensions that run all through the subarachnoid space; and then you've got the pia mater, which is a very delicate membrane that runs right next to the grooves and the bulges, the gyri and the sulci of the brain. Okay, so you can see here the pia mater, okay, this white line here that I'm tracing, just superficial to the gray matter. You can see the arachnoid mater, these web-like extensions all through the arachnoid here. And then you could see these little funny protrusions into the subdural space. These are arachnoid granulations, and what they do is they allow the cerebrospinal fluid to rejoin the general circulation, and as a result restore fluid flow through the nervous system. So remember that the cerebrospinal fluid is made by the choroid plexuses in the ventricles of the brain. It circulates all around the inside and outside of the brain and the spinal cord, and then when it's ready to rejoin the circulation, it does so at these granulations, and then what happens is the venous blood returns it eventually to the heart. You can also see that there are quite a few blood vessels that run through the brain, but even though they're in close contact with the brain, again, the formed elements of blood never come into physical contact with the gray or the white matter. Okay, they're isolated as a result of this blood-brain barrier. This happens to be a coronal section through the brain, so we're actually looking here right where the two hemispheres divide, and then down here this deep fissure, okay, is called the falx cerebri, and this is joined to the base of the skull at the top of the ethmoid bone, attached to a structure called the crista galli. And so what do these membranes do? Well, they cushion and protect the brain, and they also serve as a route for cerebrospinal fluid to rejoin the general circulation. So you've got all this protection keeping the brain and the spinal cord happy and healthy. These same three layers are around the spinal cord as well. You've got the vertebra on the outside. You've got the periosteum surrounding the vertebra, and then you've got the vertebrae themselves that enclose the membranes that surround the spinal cord, which include the dura, the arachnoid, and the pia mater. And then, of course, the spinal cord is bathed in cerebrospinal fluid, and it's also tethered both vertically and horizontally. It's tethered horizontally by ligaments that are called denticulate ligaments that radiate out laterally from the spinal cord, and it's tethered vertically by a ligament called the filum terminale. It runs from the base of the spinal cord and attaches to the coccyx, and that keeps the spinal cord from bumping against its bony protections as well. So both organs of the central nervous system are very well protected. The brain, in case you're wondering, is floating in the cerebrospinal fluid just like a goldfish in a goldfish bowl. Okay, if the brain doesn't rest on the cranial floor, it floats in this fluid. If it rested on the cranial floor, it would damage itself. The delicate tissue of the brain would begin to bleed, and that could result in an increase in intracranial pressure that could end up destroying nerve cells and could result in coma or even death. So let's talk about the meninges. The dura mater is the tough outermost layer. It has a double layered external covering that consists of the periosteum, which is attached to the inner surface of the skull, and then the meningeal layer, which is the outer covering of the brain. It folds inward in several areas, including the falx cerebri and the tentorium cerebelli. You might say, well, okay, where are those? The falx cerebri is the area between the two cerebral hemispheres, and the tentorium cerebelli is the groove that exists between the cerebellum and the occipital lobe of the brain. Okay, so a couple of areas where these membranes run inward. The arachnoid is the middle layer, and it has web-like extensions. Arachnoid means of or pertaining to spider or spider-like structures. In this case, the web-like extensions of the arachnoid mater look like a spider web, hence it gets that name. The arachnoid villi reabsorb cerebrospinal fluid and send that fluid back into the general circulation. The pia mater is the internal layer, and it clings to the surface of the brain very tightly. It's a very delicate layer, but it does allow protection for the brain as it floats in this cerebrospinal fluid. So here you can see an actual brain dissection. We're looking at the back of the head here. You can see the occipital lobe and the cerebellum, and here you can see the skull and the scalp. Okay, there's the superior sagittal sinus. This outer tough covering is the dura mater. Down here you can see the transverse sinuses. The transverse and superior sagittal sinus come together in this area that's called the confluence of sinuses, and this is just where blood that's drained from the brain is eventually going to rejoin the general circulation via the jugular vein that will eventually drain into the superior vena cava, which drains directly into the heart. Okay, so that's essentially the blood flow coming down from the brain. Cerebrospinal fluid is similar to blood plasma in composition. It's made by the choroid plexus, which are capillaries in the ventricles of the brain that filter the plasma and generate this watery cushion that protects the brain. It's circulated in the arachnoid space, the ventricles, and the central canal of the spinal cord. And I've told you that we would get a look at the ventricles of the brain. Here they are. This is an anterior view, so we're looking head on at the brain, and you can see here these lateral ventricles. Okay, so you can see one lateral ventricle, two lateral ventricles. Okay, there's the third ventricle. There's the interventricular foramen. Foramen just means hole. This is how the choroid plexus is in the lateral ventricle. Their CSF rejoins the fluid that's made in the third ventricle, which flows via the cerebral aqueduct into the fourth ventricle, which exits via lateral and median apertures to flow around the outside of the brain, and also exits into the central canal to bathe the internal spinal cord, as well as this fluid that escapes bathing the outside of the spinal cord. So there's fluid flowing within and around the brain and the spinal cord all the time. Okay, so here you can see the lateral, the third, and the fourth ventricles in the central canal. We can also see the upper and lower horns of the lateral ventricles. This is the anterior horn. This is the inferior horn. And when we look at it from a lateral view, you can see what I was saying about how the lateral ventricles look like Cincinnati Reds logos, flipped around, but you can still see. There's a lateral ventricle that's in the left hemisphere, and this is the lateral ventricle in the right hemisphere. So you can see here the anterior and posterior and inferior horns of the lateral ventricles, and you can see the third ventricle, which is shaped very much like a bird's head. Okay, you can see the intermediate mass that joins the two thalami together here, forming what looks like an eye, and the rest of the shape of the head is due to the irregular shape of the third ventricle, which is attached by the interventricular foramina to these lateral ventricles, one on either side. And then, of course, the cerebral aqueduct, which brings the cerebrospinal fluid down into the fourth ventricle, and then the median and lateral apertures, you can see them here and here, allow that fluid, some of that fluid to escape and bathe the outside of the brain and the spinal cord, while the rest moves down through the central canal. And what you're looking at here is just a map showing you how the cerebrospinal fluid is made and its route of circulation. So the cerebrospinal fluid is produced by the choroid plexuses in each ventricle. It flows through the ventricles into the subarachnoid space through the median and lateral apertures. Some of the cerebrospinal fluid flows through the central canal of the spinal cord. It then flows through the subarachnoid space and is absorbed into the dural venous sinus through the arachnoid villi, and you can see those here. Dural venous sinus receiving blood from these arachnoid granulations. Okay, you can see some arachnoid villi here and here. Okay, they're strategically located all along the area that is going to drain into the dural venous sinus. You can see here the superior sagittal sinus. This is just one of the places where the blood that's drained from the brain collects and then eventually drains down into the confluence of sinuses and will eventually return to the general circulation via the jugular, the internal and external jugular veins, which will eventually drain into the superior vena cava, which will eventually drain into the right atrium of the heart. And as long as this free flow of fluid is not obstructed, everything's fine. We're constantly generating cerebrospinal fluid. We're constantly bathing the brain and the spinal cord both inside and out with this fluid, and we're constantly reabsorbing it at the arachnoid villi. But occasionally what can happen is that we can have a block in the flow of this fluid, and this results in the newborn in a condition called hydrocephaly, where the CSF accumulates and exerts pressure on the brain if we don't allow it to drain. It's possible in an infant to survive this because the skull bones have yet to fuse, and as a result, what we can do is we can bypass this problem by, in the hydrocephalic infant, putting a shunt into one of the ventricles that will allow the cerebrospinal fluid to drain from these open fluid-filled areas in the brain all the way down into the abdominal pelvic cavity, where it'll be absorbed by the blood vessels that run through the peritoneum and will rejoin the circulation. And this shunt has to be left in for the individual's entire life. In the adult, we can't tolerate this increase in intracranial pressure that a blockage of cerebrospinal fluid would cause. The result would be massive increase in intracranial pressure, which could eventually result in the death of neurons, which could result in coma and lead to death. And so we have to keep very close watch on not just this condition, but any condition that would alter intracranial pressure, cause it to either rise or drop significantly. In either case, the brain could be in danger. And so this is one of the reasons why, for instance, we have such a heightened awareness of concussions, because if blood were to drain into the cranial cavity, that could result in an increase in intracranial pressure that could crush neurons and end up killing the individual. And so this is one of the reasons why the NFL has made such a big deal out of it. It's also a major problem in boxing and in extreme fighting. The intracranial pressure puts the increasing intracranial pressure puts the individual in increased danger of losing consciousness and perhaps dying. Okay, the blood-brain barrier is one of the least permeable biological barriers in the body. It excludes many potentially harmful substances from getting to the brain or the spinal cord, and it's useless as a barrier against certain other substances, such as fats and fat-soluble molecules, respiratory gases, alcohol, nicotine, and also anesthetics. And what do all of these compounds have in common? They all are what we call nonpolar, meaning if we were to look at the molecule from the outside, what we would find is that the distribution of electrical charge all around the surface of that molecule is basically uniform. There's not a positive and a negative end to the molecule, which means that these molecules are essentially greasy and they dissolve very well in lipids, and that means that the phospholipid membrane that forms the cell membrane doesn't provide much of a barrier to the passage of these substances into and out of the blood and into and out of neural tissue. So this is one of the things we have to be aware of. This is why, for instance, it's very easy to feel the effects of alcohol in a short period of time, because it passes through the blood and into the cerebrospinal fluid, and from there into the neurons, and it affects how they operate. Same thing is true with oxygen and carbon dioxide, primarily because of their small size, but also because of their lack of polarity. It's easy for them to pass through these membranes. The same is true for nicotine and a lot of anesthetics. They're all very nonpolar molecules. Now, TBIs are a big topic these days. Concussions, one of the major TBIs. This is a brain injury that results in no permanent brain damage, but if we experience serial concussions over an extended period of time, this can result in scarring of the brain or bleeding into the brain, which could eventually impact its function. A contusion is where nervous tissue destruction takes place, and because we're talking about neurons in the central nervous system, these neurons will not regenerate. They have no ability to repair themselves because that process, that biochemical process, is turned off in this type of tissue. That's why spinal cord injuries are permanent. That's why this type of brain injury is permanent as well. Cerebral edema results in swelling as a result of inflammation, and it can compress and kill brain tissue, which again in the central nervous system does not regenerate. Okay, remember that we only lose neurons as we age. They have no ability to repair or to multiply. All that biological response, all the information for that has been turned off in these tissues. A cerebrovascular accident results from a ruptured blood vessel supplying a region of the brain. The brain tissue supplied with oxygen from that blood source will die, and as a result, loss of function or death can result. Hemiplegia is one-sided paralysis resulting from stroke, and aphasis is damage to the speech center in the left hemisphere, resulting in an inability to speak. Individuals that suffer this kind of damage often have to relearn to talk, if they can at all, using different regions of the brain. And while we're on that topic, we should point out that the brain does have what we call plasticity as one of its characteristics, meaning that it's possible to retrain parts of the brain to do certain things, bypassing where the damage is to some extent. It all depends on how extensive the damage is and whether or not the training regimen is effective, and it varies on a patient-to-patient basis. A TIA is a temporary ischemia to the brain resulting from a restriction in blood flow that's momentary. It can often be a warning sign that more serious cerebrovascular accident could be coming. Okay, so you want to keep an eye on these TIAs. Another type of disease we should address is Alzheimer's disease. It's a progressive degenerative brain disease. Basically, neurons begin to die. It's mostly seen in the elderly, but it can begin in some individuals in middle age. Structural changes in the brain include abnormal protein deposits and unusual neuron shapes, where you see twisted fibers and tangled cytoskeletal elements inside these neurons that may impede their function. Victims experience memory loss, irritability, confusion, and ultimately hallucinations and death. And again, this is a condition that's going to vary in its presentation and its progression from individual to individual. Not everyone is going to see it end in hallucination and death. Some people may simply experience confusion and memory loss, and that might be the extent of it. Some of them may progress very rapidly to hallucination and death. What causes Alzheimer's disease? And the answer is we don't really know. Okay, we think that there may be a genetic component to it, but it's also thought that there are some environmental factors that come into play as well. It is not an inevitable consequence of old age that we would suffer from Alzheimer's and dementia. Okay, this is only seen in a percentage of the population. Okay, we're going to move now from the brain to the spinal cord and talk a little bit about its anatomy and its function. It extends from the foramen magnum, that's Latin for big hole, which is in the occipital bone of the skull, to the first or second lumbar vertebrae. Remember that the lumbar region is the lower back. After the second lumbar vertebrae, all that we have inside the spinal canal is nerves, okay, spinal nerves called the cauda equina that radiate out from the intervertebral foramina to innervate structures in the hip and the lower limb, as well as organs in that region. It provides a two-way conduction pathway from the brain to the peripheral nerves and back again. Okay, so there are both ascending and descending pathways in the spinal cord. The ascending pathways run from the peripheral nerves to the spinal cord, from the spinal cord up to the brainstem, from the brainstem up to eventually the thalamus and then the cerebral cortex. And along the way, most of these pathways will cross from the right to the left or the left to the right for reasons that we don't honestly understand completely. It's just the way it is in most higher mammals. There are 31 pairs of spinal nerves that come from the spinal cord, where the cauda equina is a collection of spinal nerves that exists at the inferior end. Okay, so you can see here the brain. Okay, there is the cerebellum, there is the occipital lobe. Buried in here is the brain stem, and then we start with the cervical spinal nerves, and then we pass through what we call the cervical enlargement, which is a slight bulge up here in the neck. And the reason for this enlargement is so that there are nerves that supply structures in the shoulder, the upper limb, the neck that radiate out from this region. So we have to make space for those nerves to protrude through these intervertebral foramina. As a result, there's the bulge. And then moving down, we can see the thoracic spinal nerves in the chest region. Below that, the lumbar enlargement in the lower back, which is the prelude to the cauda equina, the filum terminale, which attaches the spinal cord vertically to the coccyx, and the cauda equina, of course, are the nerves that radiate out from the middle and lower lumbar region that supply structures in the hip, the lower limb, and the organs in those regions as well. You can also see, looking at, this is a posterior view of the spinal cord, you can see the different meninges that surround the spinal cord. You can see the dura and arachnoid mater that surround and protect the cord, similar to the layers that are in the brain. And of course there's cerebrospinal fluid flowing all around the outside of the cord and bathing the central canal that pierces the gray matter in the spinal cord as well. Below the lumbar region, you can see the sacral spinal nerves radiating out, again serving structures, organs and muscles and such in the hip region and also in the lower limbs. And of course we've got meningeal coverings surrounding those as well. For those of you who are curious, the epidural is generally given somewhere around here because that allows the anesthetic to affect spinal nerves as opposed to potentially damaging the spinal cord itself. An epidural is just an anesthetic that's given as an injectable that blocks pain from getting to the brain for the duration that the anesthetic is inside the central nervous system, and allowing those pathways not to ascend. And that allows now painless childbirth or relatively painless childbirth. It allows surgical procedures to be done on regions at or below the hip or knee. So it's just one of the ways that we can temporarily block sensation in order to do surgical procedures. Okay, if we look at the spinal cord as a cross-section, what we find is that unlike the brain, the gray matter of the spinal cord is actually inside and the white matter is on the outside. There's a pair of posterior and anterior horns. The anterior horns are ventral horns, of course, are on the front of the spinal cord. The gray matter surrounds the central canal, which is filled with cerebrospinal fluid that flows right through the middle of this butterfly-shaped mass of cell bodies and unmyelinated axons. And then there's exterior white matter that contains ascending and descending nerve tracts on both the dorsal, lateral, and ventral side of the gray matter, where they're called columns. You've got a pair of dorsal columns, a pair of lateral, and a pair of ventral columns that carry information in a particular direction and of a particular type, either up to the brain or coming down from the brain. The meninges cover the spinal cord, and the spinal nerves leave at the level of each vertebrae. The dorsal root is associated with the dorsal root ganglia, which are cell bodies that lie outside the central nervous system, and this is how sensory information comes in to the spinal cord. Sensory information comes into the back. And then the ventral root, which allows motor information to leave the spinal cord, head out to the peripheral nerves, and ultimately to the target. And it contains primarily axons. And if we look here at this cross-section of the spinal cord, you can see these different structures. You can see here the dura mater and the arachnoid, and then of course there's a pia mater, just like in the brain. And here is the ventral root, one on each side, and you can see them coalesce into a spinal nerve, and there's the dorsal root ganglion, which will anastomose into the ventral root and form now our first mixed nerve, the spinal nerve, which can carry both outgoing and incoming information because of the layers of insulation that exist between the nerve cells that are inside the nerve. And then after this happens, after these dorsal and ventral roots run into each other, what will happen is that these nerves will split and coalesce many, many times as they run through the body, and most of the nerves in the body we'll find are what we call mixed nerves, meaning that they can carry information in either direction. There are a few that are exclusively sensory, so they only carry afferent information back into the central nervous system, and there are just a very few nerves that are exclusively motor, meaning they carry only motor information to a target to make it do something. You can see here the central canal through which the cerebrospinal fluid flows. It's again made by the choroid plexuses and the ventricles of the brain. This is the gray matter. Okay, these are the dorsal horns, these are the lateral horns, and these are the ventral horns, and these are the columns. Okay, these are the ventral columns here and here. These are the lateral columns here and here, and these are the dorsal columns here and here. And again, they carry information either up to or down from the brain and brainstem, and they only carry a certain type of information depending on what particular pathway we are addressing. And the reason that this is possible is because of the extensive insulation that exists between these tracts of nerve fibers, so that we don't short circuit on our way up to or down from the brain. And of course there are blood vessels that run all through the outside of the spinal cord, and they're there of course to supply oxygen and nutrients and keep the spinal cord functioning. Nerves and ganglia outside the central nervous system are what we call the peripheral nerves, and they make up the peripheral nervous system. A nerve is basically a bundle of neuron fibers which are bundled inside a connective tissue sheath. And so what we see here is just a look at how the brain, the brainstem, and the spinal cord and the peripheral nerves are set up. So let's start with something relatively straightforward. Let's start with this idea that we want to do something. We want to carry out some sort of motor activity. Okay, so what would happen is that in the precentral gyrus of the brain, we would receive a plan of action. Okay, that plan of action would be relayed through association neurons ultimately to the precentral gyrus, which would then, through projection neurons, send this nervous signal down through the white matter of the brain. It would run past the thalamus, and eventually it would hit the brain stem, and then at some point, either within the brainstem or inferior to the brainstem, it'll cross from one side of the body to the other. In this illustration, we're crossing from the left to the right. Okay, so this is where we're carrying our response now to the motor neuron. The motor neuron will then continue to descend until it gets to the area of the spinal cord where we're going to be at the appropriate spinal nerve to innervate the target that's nearest what it is we want to move. And what will happen now is that information will stop. The motor neuron will carry through the ventral root this electrical signal, which will ultimately reach the target, and then the muscle will carry out some kind of activity. Okay. Now the reverse pathway, for instance, if we experience a pain stimulus, the sensory receptor will carry afferent information through the dorsal root, then to the dorsal root ganglion. Okay, so there's the dorsal root ganglion, there's the dorsal root. Okay, and these are all afferent nerves. This is an efferent nerve. And then what will happen is it will synapse onto interneurons in the gray matter. That will cross from one side of the body to the other, in this case from right to left. And then these interneurons will carry sensory information up to the cerebral cortex, first heading up the spinal cord through what appears to be a lateral column here, and then eventually we'll reach the brain stem. And then from the brain stem we will head to the thalamus. The thalamus, let's say, allows this information to get to our conscious awareness, and eventually it runs through the white matter in the deeper areas of the brain to reach the cerebral cortex, generally in the sensory association area, where the brain will assign a category to the stimulus. And then through association neurons, that information will be relayed from the, first the information will reach the postcentral gyrus, which is the primary sensory cortex, and then that information will be relayed by association neurons to the sensory association area of the brain, which is in the parietal lobe. And there we will assign a category to the stimulus, and then we'll decide what we want to do about it by relaying that information to the area of the brain that's involved in higher reasoning, the frontal lobe. Okay, so that's just sort of a thumbnail sketch of how the information flows in the brain. And again, you may ask, okay, well, why this crossing from left to right and right to left? And the best answer we have is we don't know. Okay, we don't understand why the right side of the brain doesn't control the right side of the body and the left half of the brain doesn't control the left half of the body. It's just the way evolution has seen fit to wire us. There are organisms in which the appropriate side of the brain controls that same side of the body, in insects, for instance. But again, the reason that it's this way in higher organisms isn't entirely clear. So if we look at the structure of a nerve, what we see is a collection of connective tissue that's very similar to what we see in muscles. Okay, we see an endoneurium that surrounds each nerve fiber. Groups of fibers are bound into fascicles by perineurium, and those groups of perineurium-bound fascicles are bound together in a sheath called the epineurium. And why is this? This is to insulate the electrical information running through these neurons so that we don't have what we would think of as short-circuiting. Okay, this, along with the myelin, helps to keep the electrical signal going where it's supposed to go. In addition, blood vessels are included in this connective tissue, and they're of course vital for nerve function. The other thing it does, of course, is provide structural support for the nerve so that this delicate tissue isn't easily damaged. And generally, if you look in the body, the way the body's set up, what you tend to find is that a nerve and a blood vessel, both a vein and an artery, as well as a lymphatic vessel, they tend to run together in major tracts that run through the body, all surrounded by connective tissue. And the reason for that is to provide the body a better structural plan, more structural integrity. That way you don't have to route your wiring and your plumbing, so to speak, through independent openings and things like bones and other types of connective tissue. As a result, the number of weak spots in the body as a whole is minimized, and we hold together better. So let's talk a little bit about the peripheral nervous system. Mixed nerves, again, carry both sensory and motor information. Remember, sensory information is coming into the central nervous system. Motor information is heading out from the central nervous system to the peripheral nervous system. There are sensory nerves, which carry impulses into the central nervous system, and efferent nerves, which carry impulses away from the central nervous system. And you might ask, okay, how can I remember the difference between the two? Look at the word E, okay? E for exit, E for efferent, okay? So these are always going to carry information that exit away from the central nervous system, and the afferent nerves are going to be those that carry the information in the opposite direction into the central nervous system. So just remember E for exit. So let's look at the cranial nerves. These are 12 pairs of nerves that serve the head and the neck. There's only one pair of cranial nerves that extend outside this region, and that's the 10th cranial nerve, called the vagus nerve, which extends all the way down into the thoracic and abdominal cavities, and it is the major parasympathetic lead to the internal organs. Most of these are mixed nerves, but there are 3 sensory nerves that have no motor information running on them at all. And these are the cranial nerves. This is one way to remember them. Okay, there are several mnemonic devices out there that will help you remember these nerves in the order that they occur, from the most rostral part of the brainstem to the most caudal part of the brainstem. The rostral part, the part closest to the nose; the caudal part, the part closest to the dorsal tail of the brainstem. And the device given here is OOO, to touch and feel very green vegetables. And then we've got accessory and hypoglossal here. So the first letter of each mnemonic is responsible for the first letter of the actual cranial nerve. And you can see that there are 12 of them. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12. Okay. There's another mnemonic that I was taught a long time ago that's used by medical students. It's been there for... Okay, so my old style mnemonic is on the right, and the one in your book is on the left. Which one you use, really, it's up to you. Okay. Mine goes: on old Olympus towering tops, a Finn and German viewed some hops. These are all 12 cranial nerves. Again, the first letter of each word is the first letter of the cranial nerve on the brain stem. Okay, so this would be cranial nerve number 1, the olfactory. There's the optic, the oculomotor, the trochlear, and then the trigeminal. Okay, and then the abducens, and then the facial, and then we've got the auditory. This is the 8th cranial nerve. The new name for it is the vestibulocochlear. Okay, it used to be called the auditory nerve. The glossopharyngeal, the vagus, which is the 10th cranial nerve, the spinal accessory nerve, and then down here the hypoglossal nerve. All right, so just a different way to remember the same thing. I just use this because this is the one I've always used. There are a ton of them out there on the internet. Use whichever one sticks in your head the best. Okay, and again, if we look at these different nerves and we break them apart by the name, okay, we can figure out what they do. So let's start with cranial nerve number 1, which is the olfactory nerve. Olfaction means to smell. The olfactory nerve, the first cranial nerve, provides our sense of smell. It's one of the shortest nerves in the cranial nerves. It has one of the most direct pathways back to the brain, and it is purely a sensory nerve. The optic nerve is a sensory nerve responsible for vision. Okay, it runs from the back of the eyeball through the optic chiasm and eventually projects back to the occipital lobe of the brain where we process visual information. The oculomotor nerve is responsible for moving the eyeball around. And how do I get that? Oculo meaning of or pertaining to the eye, motor meaning to move. The trochlear nerve is also a nerve that's responsible for moving the eyeball around. It is called the trochlear nerve because it innervates a muscle that runs through a loop that's on the top of the eyeball called a trochlea. And so it's responsible for rotation of the eye. The trigeminal nerve is a mixed nerve to the face, and it innervates muscles, for instance, that are involved in chewing. And then the abducens nerve, the sixth cranial nerve, is also involved in abducting the eye. Okay, so it helps to turn the right eye right and the left eye left. So there's three nerves really that move the eye: the oculomotor, the trochlear, and the abducens. And then the facial nerve is sensory for taste and motor fibers that run to the face. It has 5 branches. Vestibulocochlear, or the old style auditory nerve, the 8th cranial nerve, is sensory, and it's involved in balance and hearing, but there's also a motor component to it because it controls a muscle called the tensor tympani muscle that is triggered in our response to loud noises. Okay, so it's a mixed nerve as well. The glossopharyngeal nerve is sensory for taste, but it also has motor fibers to the pharynx, so it controls how we swallow. So in case you're wondering, if you're a speech pathologist, you might deal with the glossopharyngeal, the facial nerves, and maybe to some extent the trigeminal nerve. Okay, all will have an impact on speech. And then the vagus nerve, which is one of the very strangest nerves in the cranial set, it is the 10th cranial nerve, and it goes completely out of the head and neck and goes all the way to our internal organs and controls parasympathetic input to these organs. For instance, it's important slowing down the heart rate, slowing down unconscious breathing, and speeding up digestion, movements of the gastrointestinal system. And it also innervates structures such as the bladder and the kidneys as well. So it's a very unusual nerve. The spinal accessory nerve has motor fibers that serve the neck and the upper back, and the hypoglossal nerve has motor fibers that innervate the tongue. So this is another nerve that would also be studied by individuals who enter the field of what we call speech pathology. And so here you can just see sort of a map of the cranial nerves and what they serve. Okay, so again, to repeat, on old Olympus towering tops, okay, a thin and, go down here, German glossopharyngeal down here, viewed vagus, some hops. Okay, so that's how the rhyme would go. And here you can see what each nerve does, and what they've done is they've color coded it for you as to which are mixed, which are sensory, and which are purely motor. Okay, so going by the diagram, you can see that the olfactory and the optic nerve are they're sensory. You can see here that the trigeminal nerve is mixed. You can see that the facial nerve is mixed. You can see that the vestibulocochlear nerve is sensory, but to be honest with you, there's also a motor component. Like I said, the tensor tympani muscle is innervated by this nerve. So you can put sensory with an asterisk here. The glossopharyngeal is mixed. The vagus is mixed. Okay, you can see the mixed leads going here to the internal organs. And you can see purely motor here for the spinal accessory and for the hypoglossal. Right? So a nice way for you to map. some more information about the names of the nerves. You notice that the trigeminal nerve has 3 branches. That's why it's called trigeminal. And then, just in case you're interested, the facial nerve has 5 branches. 5 branches. And very often we can pick up certain defects in the brainstem that result in either bilateral or unilateral presentation of motor defects or sensory defects in structures in the head, neck, and face. Okay, so this is one of the ways, for instance, we can figure out if there's been stroke damage to regions of the brain that innervate these different nerves appropriately, or if there's just been damage to the nerve itself. If it's just damage to the nerve, then it'll usually present on one side of the body or the other. If it's a damage that's taken place in the brain stem or the brain, it may have a bilateral presentation. Okay, there's a pair of spinal nerves at the level of each vertebra for a total of 31 pairs, and they're formed by the combination of the ventral and dorsal roots of the spinal cord, and they're named for the region that they come from. Spinal nerves divide soon after they leave the spinal cord. Rami are branches of a spinal nerve. They contain both motor and sensory fibers. Dorsal rami serve the skin and the muscles of the posterior trunk, while ventral rami form a complex of networks just for the anterior region. And so here you can see the different rami. Okay, this again is the ventral root. Okay, here is the dorsal root and the dorsal root ganglion. You can see how they run into each other to form the spinal nerve. This is in the thoracic region. There is the dorsal ramus, okay, and the dorsal ramus will give rise to nerves that serve the musculature in the back, for instance. And then the ventral ramus will generate nerves that serve the anterior and lateral portions of the thoracic region. A plexus is a network of nerves that serve motor and sensory needs of the limbs. They form from ventral rami of spinal nerves in the cervical, lumbar, and sacral regions, and there are 4 major plexuses: the cervical plexus in the neck, the brachial plexus in the upper arm, and the lumbar plexus in the lower back, and finally the sacral plexus in the very inferior aspect of the vertebral column down by the sacrum. Okay, so you can see here the different regions of the spinal cord. There's the cervical plexus here. These are the thoracic nerves. There are the lumbar nerves and sacral nerves down here. And you can see how they're numbered. Okay, so you can see that there are 8 cervical nerves. There are 12 thoracic. There are 5 lumbar, and there are 4 sacral. Okay, and here you can see what they look like. We're looking here at the anterior aspect of the spinal cord inside the vertebral column, and you can see the nerves radiating out. There are the ventral rami from the cervical plexus. Here are the ventral rami from the brachial plexus. There are no plexi in the intercostal region from T1 through T12. And then you can see ventral rami in the lumbar and sacral plexuses down here. And so what we want to talk about when we go through each of these regions of the peripheral nervous system is what important nerves we need to know from each of these nerve nets. So we'll start here with the cervical plexus. It originates from ventral rami that run from cervical 1 through 5. The most important nerve here is the phrenic nerve. The muscle of breathing is what is served by the phrenic nerve. That's the diaphragm, and it also serves structures in the shoulder and the neck. And there's an old rhyme that clinicians were taught to remember in order to keep the diaphragm operating. It goes C, 3, 4, 5, keep the diaphragm alive. The cervical spinal nerves from 3 through 5 all contribute components to the phrenic nerve, and if any of those are cut or damaged, then the diaphragm will quit working and you won't be able to breathe. Okay, so that's sort of a surgical guidepost for doctors and nurses. Here you can see the brachial plexus. It originates from ventral rami in C5 through C8 and also in T1. Important nerves include the axillary, the radial, and the median nerve, as well as the musculocutaneous and the ulnar nerve. And the areas served include the shoulder, the arm, the forearm, and the hand. And you might say, okay, well now how am I going to remember what nerves go where and what they serve? Well, think about again the different parts of the body. Think back to the map that we made in the very first week of class, and remember these different regions, right? The axillary nerve is going to be near your armpit. The radial nerve is going to run next to the radius, which is on the thumb side of the forearm. The median nerve is going to run between the ulnar and the radial nerve. Okay, the ulnar nerve is going to run next to the ulna, which is on the little finger side of the forearm. And the musculocutaneous nerve right here is going to serve structures that surround the superficial aspect near the elbow. Okay, near the elbow and near the forearm. Now we can look here at the lumbar plexus. Okay, it originates from ventral rami from L1 through L4. Important nerves include the femoral, the obturator, and the areas served include the lower abdomen and the anterior and the medial thigh. Okay, so again, how do we know where these nerves are located? The femoral nerve is going to run right next to the femur. Okay, so you can see here the femoral nerve right here. Okay, running right down next to the femur. The obturator nerve right here is so called because it runs through a hole in the pubis of the os coxa called the obturator foramen. That hole there is an obturator foramen, and that hole there is an obturator foramen, hence the name obturator nerve. Okay, and then you can see here some other nerves as well. The anterior femoral, okay, so called because it runs in front of the femur, and you can see the saphenous nerve, which runs behind the knee. The sacral plexus originates from ventral rami that run from L4 through L5 and S1 through S4. Important nerves include the sciatic nerve. Okay, you can see the sciatic nerve right here. The reason the sciatic nerve is a big deal, it runs through the sciatic notch, and it runs between some of the deep muscles of the hip, and it's one of the thickest nerves in the body, and as a result it's easily impinged, which results in loss of motor function and sensation. This is exacerbated in individuals who are obese or who are pregnant. There's also the superior and inferior gluteal nerves. The areas served include the lower trunk and posterior thigh, the lateral and posterior leg and foot, and the gluteal muscles of the hip area. Okay, so you can see the different nerves here. There's the superior gluteal, inferior gluteal, sciatic. There's the posterior femoral, again called so because it runs behind the femur. You could see the common fibular. Again, why is it called the fibular? Because it runs next to the fibula. You could see the tibial nerve running right behind the tibia. And then there's the deep fibular nerve, which again runs next to the fibula, and the superficial fibular nerve, which runs above the deep fibular nerve closer to the surface. And then down here you've got plantar branches that serve the sole of the foot. Okay, motor subdivisions in the peripheral nervous system consist only of motor nerves. They're also known as the involuntary nervous system, and they regulate the activities of the cardiac and smooth muscles and their glands. And there's two subdivisions, the sympathetic and parasympathetic divisions of the autonomic nervous system. The sympathetic division has the nickname the fight or flight division, while the parasympathetic division has the nickname the rest and digest division. So if you want to figure out what responses are produced when these different divisions of the autonomic nervous system fire, for the sympathetic division, just think about the last time you were panicked and what did you feel happening to you. You should have felt your heart rate and respiratory rate go up. You should have felt your pupils dilate, your blood sugar and blood pressure skyrocket, and also a decrease in urine output. All of these are designed to do what? To generate more blood delivery to muscles. And also another thing that happens is that we increase the oxygen and fuel content of the blood, and that gets us ready for action. Okay, so we're ready to deal with whatever comes our way. The parasympathetic or rest and digest division does the opposite. It's the division that kicks in after a big meal, and it increases GI motility, it lowers blood pressure, it increases the filtration of the blood, so what we have is an increase in elimination of body fluids. It also constricts the pupils, and in general it's the division that's responsible for basically shoring up our body's internal fuel reserves, our fats, our glycogen, and so on. And so here you can see a chart showing you the differences between the somatic and the autonomic nervous system, the difference between the nerves, the effector organs, the subdivisions, and the neurotransmitter used. Okay, this is an excellent chart. Star this in your chapter and know this material. This is a nice summary of the differences between the two nervous systems. And this diagram here sort of shows you how the different nervous systems are anatomically set up, right? The somatic nervous system is made up of a single motor neuron that carries efferent information all the way to the target, and the acetylcholine is the neurotransmitter. In the autonomic nervous system, you have a two-neuron pathway between the spinal cord and the target. In the case of the sympathetic division, in the preganglionic neuron, acetylcholine is the neurotransmitter, while in the postganglionic neuron, norepinephrine is the neurotransmitter, and the targets are different than in the somatic motor system. They include nerves and glands and smooth muscle. And down here in the parasympathetic division, both the pre- and postganglionic neuron use acetylcholine, and their actions are generally opposite those of what the sympathetic division innervates. And they hit the same targets, but they might hit them at slightly different anatomical positions. Now, the one unusual aspect of the autonomic nervous system is shown here. Okay, what we're looking at is the this setup for the sympathetic division, where we have direct innervation of an endocrine organ, which is the adrenal medulla. And what happens when this occurs is that instead of generating now a postganglionic neuronal response that results in the release of neurotransmitter at a target that causes some sort of action to take place, we release epinephrine directly into the bloodstream, and that causes the effects of the sympathetic nervous system to last longer than they would if this response wasn't present. So this is one of the reasons why when you panic, you don't calm down right away, because you've still got tons of adrenaline in your bloodstream, accelerating your heart rate and respiratory rate and jacking your blood pressure and so on. Whereas the effects of the parasympathetic division can be quickly ended in response to a panic situation, because there's no endocrine organ that complements the activity of the parasympathetic division the way that the adrenal medulla does for the sympathetic division. So if we look at the parasympathetic division, the preganglionic neurons originate from the craniosacral regions, and they include cranial nerves 3, 7, 9, and 10. They pop out from S2 through S4 in the spinal cord. Okay, so those are the sacral regions. Due to the site of preganglionic neuron origination, the parasympathetic division is also known as the craniosacral division. Okay, the reason for that is that a lot of the nerve leads come up from near the head and the neck, whereas or the rest of the nerve leads come up down near the sacrum. Okay, so craniosacral division, two-neuron pathway for this autonomic division. The terminal ganglia are at the effector organs, and the neurotransmitter in both the pre- and postganglionic neurons is acetylcholine. The parasympathetic division is responsible for housekeeping activities. It conserves energy and restores fuel. It maintains necessary body functions. And remember the capital letter D for its tasks. It's important in digestion, defecation, and diuresis. And here you can see the setup for the parasympathetic division. Okay, you can see the parasympathetic leads from the cranial part of the division heading to the eye, the salivary glands, the heart, and the lungs, and the stomach, pancreas, and all these other internal organs. They're all innervated by the vagus nerve. Okay, so this is all vagal nerve leads. And then down here near the sacral division, I've got some sacral nerves that target the bladder and the genitals. Now in the sympathetic division, it's called the thoracocolumnar division. And you can see here in this same diagram, it has a slightly different anatomical setup. Most of the nerves from the sympathetic division originate in the thoracic region of the spine and the upper lumbar, and they hit paravertebral ganglia that are very close to the spinal cord, just outside the vertebral column. And from there, a second neuron will carry the sympathetic information all the way to the target. So you have a short preganglionic and a long postganglionic neuron that head all the way to the target. And you can see here that a lot of the targets are the same. The eye, the skin, the salivary glands, and then down here the lungs, the heart, the stomach, the pancreas, liver and gallbladder, and so on. Okay, all the way down to the bladder and the genitals. And what are the effects? The effects are generally opposite those of the parasympathetic division, the craniosacral division. So what we tend to see is an increase in heart and respiratory rate, an increase in blood pressure and blood glucose, dilation of the pupils, and secretion of adrenaline into the bloodstream. And this readies us now for a fight or flight situation. In the sympathetic division, the preganglionic neurons originate from thoracic 1 through lumbar 2. The ganglia are at the sympathetic trunk, which is very near the spinal cord. There's one on each side of the vertebral column. Short preganglionic neurons synapse onto long postganglionic neurons, and these transmit impulses from the central nervous system all the way to the effector. The neurotransmitters include norepinephrine and epinephrine that hit the effector organs directly. And what you're seeing in this next diagram is just an illustration of how these autonomic sympathetic pathways get out of the central nervous system and head to their targets. There's one of several routes that they can take. Okay, if we look here at the example of what we call a splanchnic nerve, which passes through the paravertebral ganglia and synapses on a intramural ganglia that's outside the paravertebrals, what happens is that we head out through the gray matter of the spinal cord, through the ventral root, and then we hop directly through the paravertebral ganglia through what are called these rami. Okay, and essentially what happens when we exit is that we are going to exit through the white ramus and then head on all the way down to our intramural ganglia, and that from there the second neuron will go all the way to its target. But those are the exceptional sympathetic nerves. Most of them are going to synapse inside these paravertebral ganglia, and then the second neuron will head all the way out to the target. So here you can see an example of an outgoing nerve. Okay, we're going to follow this green pathway here. It's going to head all the way around, and it's going to synapse at the level of this paravertebral ganglia, and then the second neuron will exit through the gray ramus and will head all the way now to its target. Okay, so that's the white ramus, that's the gray ramus. Or we can take another pathway. Okay, we can head out through the gray matter of the spinal cord, through the white matter, through the ventral root. Okay, we can enter via the white ramus, and then we can either head up or head down within the paravertebral ganglia, synapse onto a second neuron, which will exit through the gray ramus and then head all the way to the effector and then cause a particular action to take place. The sympathetic division has the nickname the fight or flight division. It responds to unusual stimuli. It takes it over to increase activity such as heart rate, respiratory rate, blood pressure, blood glucose, and so on. Remember E in this division. It's responsive primarily in situations such as exercise, excitement, emergency, and embarrassment. Now, the nervous system comes during the first month of embryonic development. Remember that we go from the morula to the blastula, and then we form what's called the embryonic disc, and then on the top of that embryonic disc, the primitive streak forms, and that gives way to the neural tube, which drops down below the surface of the ectoderm, and then we begin to close that groove up from tail to head. The head part's eventually going to form the brainstem and the brain, and the rest of it projecting below the brainstem is going to be the spinal cord. Any maternal infection can have very harmful effects on nervous system development in this period during the first trimester. Hypothalamus is one of the last areas of the brain to develop. No more neurons are formed after birth, but growth and maturation continues for several years afterwards. The brain reaches its maximum weight in a young adult, and then it begins to shrink thereafter. And why does this happen? Because neurons die. And as neurons die, brain function is slowly lost, and that is responsible for some of the loss of mental sharpness and confusion that is seen very often in individuals of advanced years. Okay, that brings this talk to an end, and I will see everyone in class.